Japodi, japodi, jop, 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 jop. No niin, mennään tonne missä ollaan. Ja saanko show-valo, kiitos. Mitä? Voisi olla paneelivalo, koska niin kuin, otetaan ihan koko stage. Noin. Yes. Se on makuasia, haluatko sen noin päivän?
Joo, no mä kääntelen sitä.
Yes. Okei. Ole hyvä.
Mä teen siihen siis tosi hiljaisia ääniä ja siis mä hönkäilen. Niin mä hönkäilen tänne sivuun, että se ei ota sitä suoraan, kun ei ole tuulisuojaa, mutta Mä teen siis tämmösiä ääniä. Tämmösiä ääniä siihen. Toi on ehkä kovin ääni, mikä. Se ei poksu nyt. Ei poksu. Okei. Okay. No niin, tää riittää mulle. Okei.
Mitä sä oot tehnyt aikaisemmin, niin sulla ei siinä jonkin verran kaikkea.
En muista, ei välttämättä. Se voi olla aika kuiva.
Okei, selvä.
Joo. Ei kosketa.
Hello everyone, I'm Zam. Seed when they share seed, busy colonial histories from parts of Asia, parts of Europe.
Okei, Hanna, mikä tämä nature on sillä tavalla, että meillähän todennäköisesti nyt meillä on aika vähän ihmisiä, joista ollaan ton takia sisällä. Mutta siis se, että onko ajatus se, että pyritään tekemään mahdollisimman vähän täältä, vai saako mun pistää vähän lisää potkua tuon, mitä tulee ulos?
No siis lähinnä silleen, että ihmiset kuulee, mitä mä teen. Okay. Joo.
Otetaan vielä vähän. Ota Matti vähän ylös. Hanna ylös. Katsotaan vähän, että semmoinen, että...
Niin. Se ei tule päälle sieltä. Joo. Sitten mulla on siis muutama tommonen, etteihän ne mene liian kova. Joo. Tää on sitten ehkä vähän kovaa. Kun mä siis puhun ja hönkäilen, no niin. Tulee pyörälimat tuolta keuhkoista. Joo, mutta siis onko tässä tarkoitus, että kaikki puhuu oman presentaationsa myös mikkiin? Okei. Okay.
Koska mennään striimiin niin paljon.
Joo, just niin.
Yleisö on pääasiassa tuolla maailmalla jossain.
Just näin. Mä tarviin jonkunnäköisen standin. Mä voin... Toi on se slaidikone.
Pavel! Would you please take as a presenter?
Yeah, I need some sort of a stand because I have my notes.
Mic stand, or you mean?
A note stand.
Yeah,
so
it's this one is the meant for that.
Okay, so the mic should be somewhere near, or
Usually we keep mics in hands, or we have Madonna mic, which can be.
Aha, okay. So I'll do so that when I speak, I have the mic in my hand.
Yeah.
And when
Yeah.
Okay, we'll do so.
Is it okay?
Yeah.
And is it okay for you to stay here, or you want this to be?
Yeah, it's just I would prefer if I have a note stand for my iPad while I sing and breathe because.
Yeah, we can. So we can put this in the middle too.
It has
It's like-
wheels.
yeah.
Yeah. Okay, that's great. So
And we can also move.
I'd be kind of in the middle of this and yeah.
Maybe we just not using it. Yeah, but I mean maybe we are just taking it away. I mean, not usable. Yeah, but we need to take it away.
Okay.
It's not usable.
Okay. Right.
Yeah.
Maybe just.
That's
Do it yourself.
Okay.
And you can just put it like you put hood. Sound a bit just for.
Okay,
You can say so.
check. Hello.
Yeah. Is it good?
This works.
There is no too much attack. Yeah.
Okay.
So let's discuss. You can speak. So.
Let's
you
discuss
will be-
something. Let's try at the
And
front.
you have your presentation, right?
Yes, it should be somewhere there.
And the presentation is here.
Yes.
How were you this morning?
Tired.
Okay. How, was it hard to get here?
I took a cab, so no, it's well with the bus strike, so it's pretty difficult. But we booked a cab, so
Yes. Okay. Pavel, now that
Yeah.
so
So we would have it a bit
lower.
a bit lower,
So
like
You feel
because
okay?
basically the,
Yes.
the air when speaking
Yeah,
with
yeah.
the
Yes.
Yeah, I'm telling it to Yasmin.
Yes.
So basically we are having a conversation. Hey, so okay, how was the taxi ride?
that was
Ru-
fine.
rush hour?
Actually, no, it was pretty quick. It was only 15 minutes, and a lot of snow. I don't know if that made any difference, but surprisingly good.
Much better. Not so many anymore.
Yeah,
Because
yeah.
It's lower, so it's
Let's so lower, like it would be something like this, and yes. Okay.
So about the presentation, we also have this. So
Yes.
if you feel like more comfortable working here, you can use this to change your slides.
Yeah.
Tämä keltainen on sun, se on tossa. Ei vielä, mutta se on sun ja tasa. Joo. Se on sun ja sitten kun on valmis, niin tämähän on elossa, eikö vaan?
Hello everyone.
minuuttia, 2 minutes.
Nina, are you gonna introduce all of you?
Yes.
Yeah. Okay.
As you before you go on.
Okay, great.
Yes.
So, good morning. Welcome to the third day of Kuva Research Days. My name is Mikaelo, Professor of Artistic Research. And today we have a day program put together by postdoctoral researcher Nina Liebenberg. And unfortunately, today is a bit special day in terms of traffic. So we have clearly less people live here, which is pity because we have also a very interesting and detail-full display, a small exhibition here in the space, which is impossible to make accessible online, but you will be able to follow the talks online, which is good. So I will give the mic over to Nina, so that Nina can present the day's program and the speakers. Once more, welcome.
Oh. We'll take this. Okay. Tervetuloa. I didn't expect the Mercury retrograde to wreak as much havoc as this. But alas, if you don't believe in astrology, you can start believing now. All transport and everything is down in Helsinki. But I'm very glad to be here and to have a few people here with me as well. I guess I just want to do a short introduction. This is kind of the part of the day that I left for last, and alas, here I'm standing without my script. So let's see how this goes. The day is termed Object Studies, and I hope most of you have gone through the program a little bit before coming here. And it's looking at human-disease-plant relationships. And I first want to make a little bit of a confession, actually, because when I was thinking about my introduction and actually putting this display on and putting together the program where I'm kind of very instrumental, every now and then I show my face and I do something, I realized that it's not about plants, it's not about disease, it's not about humans, it's not even really that much about objects. It really is about curatorship. And curatorship is my methodology. It's the thing that I test constantly. And it's the thing that allows me to generate meaning in the way that I need to generate meaning on a daily basis. So this program is a very sensitively, hopefully, testing curation. It's bringing together quite a diverse, strange group of people and objects. And when you see my intervals, I'll even make it more strange with the curation of even more things that I'm bringing into play. But curatorship is a wonderful methodology because firstly, it allows you to bring together all these different strands of things. And it allows you to play with them in different ways and in different relationships. And while you're looking at something, to keep something else that is in close proximity constantly in your mind. So it's always implying something beyond what the object is showing you. And now I'm backtracking and losing my train of thought. So my background is curatorship, and I did teach curatorship at the University of Cape Town for about 10 years. We started a curatorial program there in 2013, where we really wanted to test whether we could do it as an interdisciplinary program. And we welcomed in students from chemistry, anthropology, history, film and media studies. So they were coming from all over the place, from all the different departments in the university. And we found that it was quite hard to bring them together and to make them speak and make meaning together through curatorship. So we realized that within every department, there is images and there's objects, and these form the focus of a lot of the disciplines. The herbarium specimens are pivotal to the study of plants. Studies of astronomical images, slides of geological slices of rocks. Those are material and objects that researchers work with across departments. So we realized that bringing together these students and asking them to focus on objects was a way to actually run this program. And the students came, and they came with their objects in hand, and we started developing the program, and I was able to see firsthand the power that objects, through the curatorial, has to work in an interdisciplinary, cross-disciplinary way, to generate meaning and to expand dialogues and to highlight blind spots that we all sit with in our disciplines because we read the same things, we attend the same lectures. So, I hope I've put a big disclaimer into why we've got this diverse group of people that's coming to speak to us today and sing to us today, Hannah. And I would hope that within the interval that you would take time to also look at the display, because things that are talked about are popping up on the display tables. They will pop up in the displays. People that are presenting their practices will pop up in the displays on the table. So you'll find yourself there. There will be an aspect of familiarity, but also an aspect of strangeness, which I think is that perfect little uncomfortability that we need to put ourselves in constantly. So hopefully this day does that. I put the slide up, which I realize now. So there is a curator in London that did an exhibition called Home and Away. I think his name is Jeffrey Kipnis, and he described curatorship as a round trip. And I'm going to quote him on what he said to just kind of give you a sense of what it can do. In his essay Away from Home for the Wexner Centre for the Arts exhibition with the same name, featuring artists dealing with issues of home, travel, exile and sense of place, Jeffrey Kipnis writes about the concept of the round trip and how its basic outline, home, away, back, finds its most fertile incarnation in the sonata of classical music. And I quote, "The general form of the sonata is exposition, A, development, B, and recapitulation, A," with that little small thing. The first section, the exposition, introduces the sonata's melodies and themes, i.e., its cast in the home key. And then, as the exposition comes to an end, the players depart from that home. In the second section, the development, the themes endure a variety of musical adventures as they pass through a wide range of different keys, the musical expression of different locations. Key changes in the development distant from the home key are termed chromatic, meaning colorful and making the music feel exotic. Finally, in the third section, the recapitulation, the musical material returns to be repeated in the home key, just as it started. Never again to leave, yet never again to be quite the same. So I hope that the objects and the people and you that I've brought all the way from home to here, and when you return home, you will find that you've learned something and it somehow changed you in some way or form. So that's my introduction. And now how are we on time? Okay, so I'm going to introduce the first speaker, Jasmin Kempinen. Oh, where are you? I don't want to misrepresent you, Jasmin. Okay. So Jasmin is at the University of Helsinki, and she's generously allowed me and students to visit her and her lab and learn about her research. And it was so interesting, I really just thought that you were the perfect person to bring back here to chat today. So Jasmin Kempinen earned both her BC and MSc degrees at the University of Helsinki, specializing in plant physiology and development. Her current doctoral research explores the role of a receptor protein, GHR1, in the complex world of somatal immunity networks. Her core interests lie in unraveling the mechanisms behind pathogen perception and the orchestration of various signals that drive stomatal movements. Jasmin's passion for the botanical world extends beyond the lab, as she also cultivates, paints, and photographs plants. Okay, welcome, Jasmin. Looking forward to it.
Okay, thank you so much, Nina, for the invitation and the really nice introduction. So I don't really have to do that myself or too much in detail. So thank you so much. I'm really, really excited to be a part of this project and also to see all the cool looking objects you have put there for us to see today, and also maybe to look at my own research in a slightly different way after this. So, yes, my name is Jasmin Kempinen, and I am doing research at the University of Helsinki as a doctoral researcher. And my group, Maija Sirla's group in plant environmental signaling, we're interested in how plants respond to environmental signals and basically on a molecular level. So it's a different world than what we can see with our bare eyes, and because of that and all the complexity, the first part of my presentation is more like has a maybe educational tone. I hope that's all right, because I really want to explain the mechanisms first in order to fully kind of understand and really see why plants are so cool, because I think the more you know, the more interesting it becomes. So I'm studying plant pathogen interactions and stomatal immunity. I am not a plant pathologist, even though I'm interested in disease dynamics and the signaling networks. These were the objects of mine for today. And the first one on the left is a Petri dish with some growth media. It has the white speckles. They're bacterial colonies, like the white speckles you can see here, produced by a Pseudomonas syringae, a plant pathogen. And unfortunately, I couldn't bring the physical Petri dish here today. I really would have wanted, but since it's a GMO strain, it would have been kind of unpractical, and well, the law is the law. So, but at least I still want to talk about it because it's very central to my research, and it's an interesting pathogen. The other one on the right is the stomatal complex of Arabidopsis thaliana from our model organism. And this I will also explain a bit later. What is a stomatal complex? What's that all about? Although Nina already kind of introduced me, I kind of briefly want to mention something about my background and about my master's and my current project. So I did my master's in plant physiology and development, and I was mostly interested in photosynthesis and photosynthetic regulation and oxidative stress. And I was really into these tiny organelles called chloroplasts. You probably heard about them. They are the sites for photosynthesis. So I was looking at those. And nowadays I am doing my PhD also at the University of Helsinki, and I'm studying stomatal immunity, and particularly this receptor called GHR1, which I also kind of want to introduce very briefly. Even though these seem pretty different, these two topics, they are actually fundamentally somewhat related. So they both are core processes in plant function, since photosynthesis is the site for carbon, assimilation. Like, how does carbon dioxide turn into sugars, like something from a gas into building blocks of life that we also use for our energy. And they're also both very fast regulation events, like photosynthesis and stomatal function and regulation. These happen on a seconds to minute scale. So at least for a plant, that's very, very quick. And most importantly, for me personally, is they're both related to plant signal perception. How does plant interact with its environment, whether that is light for the photosynthesis or whether that is microbes and other organisms. And of course, they were both conducted on Arabidopsis thaliana, and that's something I will introduce now. So that's our model organism in plant sciences and plant genetics. It's a member of the mustard family, distantly related or related to kale and cabbage, canola, very important crops for our food and energy production. But Arabidopsis itself, it's quite modest looking plant. We can actually find it from the nature in Finland. So if you see a tiny white flowers during early spring, that might as well be our model organism. And what is a model organism? Why are they used? So it comes into practical reasons. Like Arabidopsis thaliana can be thought as kind of like equivalent of a mouse or rat. That's the model organism that animal biologists use in their research. And plant biologists like to use Arabidopsis because it's also like a mouse. It's compact. It has a somewhat compact genome and a compact size, and also short life cycle. Arabidopsis self-pollinates, which means we don't need to do any cross-pollination. It produces seeds itself. And many molecular tools are optimized for Arabidopsis. We can easily add or remove genes, and the gene removal is actually something that kind of people at least in my kind of research field use a lot, and this is conducted on this kind of knockout mutants. That's what they're usually called, and they usually lack one or several genes. They're kind of knocked out, and that's why they're called knockout mutants. And here's an example from my master's in which we got these knockout mutants for different genes. So each of them lacks a specific one gene or two genes, and they look pretty different. And from these we can compare how they look to this wild type called 0. That's our normal kind of healthy plants having all the genes still intact. And these are lacking something. So we can get some kind of hint, kind of like in a kind of like reverse way, like what would this gene do? And I've been talking about stomatal complex, but now I actually want to explain what that is. So stoma, it's the singular word for stomata, and it's from the Greek word meaning mouth, and it's makes sense if you look at this stomatal aperture and the stomatal complex. They look like tiny mouths, actually. And they are evolutionarily conserved in plants. The stomata appeared in the fossil record something like 450 million years ago when plants started to conquer land. So they are very fundamental for living on land, at least for plants. And their most important function is gas exchange and transpiration. And even like as animals, we're mostly interested in this oxygen release part of gas exchange because we breathe oxygen, we need that. But most important function of stomata in the gas exchange for plants is it's acquiring CO2 for photosynthesis. And with such a crucial task, the stomata can and must be adjusted so they can move and respond. And it can happen rapidly, as I mentioned. And last weekend I was playing around with my new digital microscope, and I took a time-lapse video of a houseplant of mine. It's from a very popular houseplant called Tradescantia zebrina. In Finnish it's called juoru. So you might know how it looks like, but it's not the best quality, but let's see if I can put it. Yes. So this is happening within 2 minutes. It's sped up. It's not it's a kind of subtle movement, but if you pay attention to this cell, you might see some small closure. At least I hope so. So they really do respond quickly, and this was a quick test, but even then I could see something, and that was really, I got super excited about that and really wanted to share that. So this happens according to different environmental signals. And I think this happens because I moved the plants from under nice grow light into darkness. So I think that might have been the signal. And indeed, some of the signals that open stomata include high light, optimal light and high humidity, whereas like darkness and drought closes. And events can happen from the signal perception to closure within minutes. So it's fast. And some of the mechanisms for stomatal closure, I want to quickly explain. I wonder if I can put the presenter view. Yes. Sorry about that. But yes, to understand why they move and how it happens, we need to look at the anatomy. So you probably see the stomatal aperture, the pore. It has 2 surrounding cells, and these are guard cells. And they are very important for the movement because they actually adjust the cells and themselves and the stomatal pore. So this is quite well studied because it's so important. It's complex and convoluted, and we have all these proteins and channels and everything. It's complex, but actually the fundamentals of it's quite simple actually, and it has to do with osmosis and water flow. So a simplified diagram kind of shows the 2 surrounding guard cells and the stomatal pore in between. So when the stomata opens, the cells are getting swollen. So water goes into the cells, and when the stomata closes, the water is escaping. So this is plants achieve this by moving solutes outside or inside from the cell. So that's the basic principle, and it's actually pretty much the same thing that happens when you're preparing, for example, an eggplant for roasting or doing tzatziki from cucumber. You want to kind of slightly dehydrate your vegetable. So you sprinkle some salt, that's the solute. You can see water coming out. That's osmosis. So that's the basic principle, which is quite simple actually. So I said that stomata can close or open to different environmental signals, and stomatal closure can happen as a part of plant defense, and this is kind of where my PhD topic comes in. And like I said, this is the CO2 capture is the important function of stomata, but this obviously requires the pores to be open. So this makes a kind of like a dilemma for the plants, since the open pores makes the plants vulnerable. They lose water, they might desiccate, but this also can allow pathogen entry, like seen the tiny microbes here going into the stomatal pore, and this can induce stomatal closure because the plants can detect this. So my question is, how do plants recognize pathogens? How do plants recognize anything really? And this is a central idea in immunity altogether, whether it's plants or animals or anything. How do organisms recognize something that is non-self? That's happening on a molecular level as well, and it's very dependent on these receptor proteins. And they kind of work like a lock and a key. Their receptors are sitting on the membrane of a cell and detecting fragments or molecules from some kind of outside invader, something non-self. And this beautiful illustration is a watercolor painting by David Goodsell, and it's depicting two human immune cells. These are the cell membranes, and these are the receptors. And you might see there's a tiny red dot inside, and that's a fragment of a virus. And the receptors are communicating through recognition of that, binding that, and then all sorts of events happen inside the cells. And this kind of innate recognition is genetic, and it's happening in both plants and animals, and it's evolutionary conserved. So the main difference between plant and animal immunity, there's a kind of core elements that they share, but the biggest difference is that plants lack mobile immune cells. Like we as animals have all these complex immunity events happening, all the like white blood cells and the T cells and everything, but plants don't have that. So that means plants must recognize them all over the place, like the recognition must occur in every cell throughout the plant, and this makes plant membranes full of receptors and kind of chemical warfare. And guard cells are particularly sensitive, since they are the kind of like gatekeepers inside the plant. And if we zoom even closer, looking at the kind of stomatal complex, this is my working model for my PhD. Don't worry, it looks convoluted, but just wanted to show that there's also receptors that recognize pathogens, and some of the components are very, very well studied. But just to highlight that this is the GHR1 that I'm studying, and that is not so well understood. And if we zoom even closer, we might see something like this. This is a receptor, how it, the protein model of the GHR1 becoming named from a guard cell hydrogen peroxide resistant 1. I didn't name it. I don't know why it's called like that, but these are how the proteins are usually named after. And we know something about it. We know from, remember those mutants that I was talking about? So these have been used to study how the plants breathe, and there's these kinetic curves of kind of gas exchange, how much plants evaporate, and this called stomatal conductance. And you might be able to see the purple line here. That is how this mutant, GHR1 mutant lacking the gene and the protein, breathes, and it doesn't respond to night or day. So it's darkness and light. It stays flat, whereas the wild type normal plant is responding and closing during the night. So something is happening. So it is needed for something. And in its role in plant immunity and kind of like microbe interactions, it's not examined at all. And this is what I'm interested in. I would like to show that it's also involved in biotic kind of interactions. So we went all the way into the kind of molecular workings and protein structures and everything, but now I want to come kind of back to the real world, because it's pretty abstract in a way, at least for me still. So how do we actually study something like this? What kind of tools we can actually use? So now I come to my other object, not physically here. So that's the plant pathogen. It's also kind of like a model pathogen. This particular strain is extracted from tomato. This is called a pathovar, not like a cultivar, but pathovar. It's a strain of pathogens. And Pseudomonas can infect various plant species. It's not like very specific to Arabidopsis or tomato, but it can infect many species. And the bacteria itself looks like this. As you can see, it has these two flagella, that's plural. So it uses those to swim around, so it requires water to move and infect. So that's how it looks, and these flagellum pieces are important for the recognition, as you might have been able to see from the schematics. And indeed, it uses stomata as entry point, so it really swims towards the pore and is able to recognize it somehow. And some of the disease symptoms: this is a plant I infected by spraying. It develops these kind of water-soaking lesions filled with fluid, and also becomes kind of like a chlorotic and yellowish. And we can extract the bacteria again and measure how well it grows in our different lines. And the interactions, it's actually pretty complex, not only on the molecular level, but how the disease is kind of developing. So it's dependent on time and kind of a pathogen, different stages of life cycles. And different stages, the bacteria wants a different thing. At first, okay, and also the plant. So first, when the bacteria comes, the stomata close, but the bacteria are also able to reopen the stomata by secreting these kind of toxins. And then plant obviously wants to kind of counter-attack this, but in time, the bacteria wants to close the stomata to induce this kind of water soaking and the accumulation of water. And again, plants would want to open the stomata again. So this is kind of like if you look at in a temporal manner, it's a pretty complex dynamics, and it's not well understood, like when and how they do these. Both organisms make the decisions. So it's an evolutionary arms race in which the host makes an adaptation against a pathogen, and the pathogen has a counter adaptation, and it goes on and on and on. So this is kind of like, you can see it in like a predator and kind of like prey dynamics, but also here. And how to study stomatal responses. I hope I'm still in time. Maybe. That's good. So how to study stomatal responses, and we have the direct methods. Really just look at the stomata and different times. And this is actually some of the related to other object I brought. I brought a microscopic slide from this experiment. So we can treat the plants by spraying or some other way, and then we take the samples with this. We use this dental paste that solidifies within like 10 seconds, and that's really important because, as I showed, the responses are super fast. So we need to be really quick also in sampling. So when we create this kind of like a mold for the plant surface, we preserve the exact moment because it solidifies pretty quickly. Then we can create a positive cast. I hope that's the kind of correct term for it. So with a nail varnish, because it's transparent, we can then see it in a microscope. And this is hopefully something that you might be able to see through the microscope. It's kind of like a topographical map of the plant surface. So these are not live cells, they are the shapes of the plant surface. Another method is extracting the epidermis. So that's the outer cell layer. And that's, we can peel the epidermis off and do the treatments in an incubating buffer, and then directly put it in a microscope slide and look at the samples. And this is, in a way, really nice, because then we can see live cells. This is not a kind of like it's not a cast or anything. There are living cells, and you can maybe see the chloroplast also here, the green speckles. So it's a one cell layer, thin strip of epidermis. So that's pretty cool. And we can go even further, and we can fix this and stain it with a fluorescent dye to make it even clearer to see. Then we can use more advanced microscopy techniques. It's, for example, confocal microscopy. And usually for these, we are interested not only in the stomatal aperture and behavior, but also some protein of our interest. So we can tag our protein of interest with a fluorescent protein. So this requires gene modifications and some gene transfer. And in this, we can get a lot of different information. We can get, depending on the fluorescent protein we used for tagging. So that can be localization, the function of our protein, interactions, and this is all in a spatiotemporal manner. So we can get information both in space and time. So I think that's pretty neat. And it also, I'm not going too much into detail here because I'm not a confocal expert. But I know that by using different fluorescent proteins, you can get different type of info, and that's fascinating. Then we have indirect methods, and you saw this kind of kinetic curves of the plant breathing. That is the gas exchange, and this we get from the gas exchange measuring machines, let's say. So this we can do from a single leaf level or a whole plant level. And these chambers that we have, we can fit a whole Arabidopsis plant here and measure the gas exchange from a whole plant level. And from this, we cannot see the stomata. We cannot know anything kind of like spatial information, but it's temporal information. That's really valuable because we can see how plants breathe within minutes or hours or even days, and we can use different types of treatments to see how this gas exchange is changing. So this is my last slide, and this is some like final thoughts that people usually, when they're talking about plant science, they start with. But I want to leave you with these thoughts because I think they're really important key points, and maybe to see and think plants in a different way. And I think plants are really sensitive and they're excellent problem solvers. And that is why they are so interesting. So they are such good problem solvers because they are sessile. They are rooted to one place and they cannot move. We as animals can escape a stressful situation, plants cannot. And this means they need to be prepared for everything and anything to survive. And this makes them so complex and dynamic. And this is kind of like one of my key interests at the moment, and a big question. I don't know if it can be ever answered. But how do plants make decisions with so many environmental signals and factors? Like, which signals do they prioritize? If they have highlight and sunny day, but the pathogen comes, do they continue photosynthesis and do they keep the stomata open? How do they make decisions? So, and what can we learn from plants? Since they are such a kind of like a elegant problem solvers, so I think they might hold many answers to problems that we are struggling with. And we need to understand them, not only to protect ourselves and our food production, but we need to understand how plants respond in a changing multifactorial environment, also to protect our environment and endangered species, but also our own food production, because we really rely on plants. So by this, I want to thank my group. There's a QR code if you're interested to see what kind of research our group does. So you can check that out if you want to see and learn more. And I also have an Instagram account if you're curious about more botanical content. Also my own paintings, watercolors, but also pictures of my plants and some scientific content and stories as well. So yes, thanks a lot for your attention. And if you have any questions, I'd be happy to answer them. Thanks.
Kiitos paljon, Yasmin. I'm pretty astounded how you managed to walk us through such complex research, and you did it really sensitively and, you know, gave a generous amount of information, but kept us with you constantly. And
I'm happy.
I cannot believe it. We've got time for a few questions, maybe one or two, just to kind of keep time running in the right direction. Does anybody have burning questions to ask Yasmin?
Hi, thank you. I was wondering, do plants then close the stomata like one at a time, or like do they open in the whole plant at the same time, or are they also somehow are they synchronized or not?
That's an excellent question. So whether the stomata are synchronized or not, and do they close individually or as a kind of like a group? So we don't actually know. It depends. Like we can see like a team effort, for example, from these gas exchange measurements. That is a effort from many stomata. But there is some like variation between like from one leaf to another, because they might have like different environmental conditions. And for sure, we know that there is some kind of like systemic signals in plants. So there are kind of like internal signals from the plant to stomatal complex to close or open. So, but in a way, there is still this kind of spatial variation within, like one leaf might be doing the gas exchange happily with open stomata, and another leaf might be more closed. Like, this is something that people are trying to understand how coordinated it actually is. It's for sure it's not like all or nothing unless it's a very strong signal like drought. But in a kind of like fluctuating kind of conditions in nature, there is a lot of variation. So
yeah, one would think, because like a bacteria can be on one leaf but not another. So
yes, and also depending again the time, like there might come there is an actually signals happening from the site of the recognition of bacteria going another parts of the plants. So there is this kind of priming for immunity. So the other parts of the plant might be more ready to protect themselves when the bacteria and the pathogen comes.
This is fascinating.
Yes.
Yes, Maria. We'll bring you a mic.
Thanks, Yasmin, very inspiring. I might just step a bit out of your explanation, because I'm very curious also about, at another level, crystals and sugar. And I know that sugar plays also a role in stomata when there is open or closed, and starch. So I don't know if you could also explain a bit of that. I don't know if it's like related with, you know, your researching precisely, but maybe you can illuminate us a bit anyways.
That's a really nice point. Like sugars indeed play a important role in the stomatal function and whether they are open or closed. And this is actually, it has been a hot topic in stomatal research. Honestly, this plant energy metabolism, like the carbohydrate metabolism, is not my strongest field, but I do know that there has been this kind of some different opinions what the sugars do and what's the role of sugars in the stomatal aperture. So is it also an osmolyte? That's one thing. Is it driving this kind of water transport in and out, like, or is it a signal from the photosynthesis? I mean, if there's a nice light environment and the chloroplasts are photosynthesizing, producing sugars, so is that a signal for the stomata to do something like close? Okay, we're getting, if it like it saturates, it maybe stomata close, or so. In a way, the starch degradation and the different types of sugars play different roles, as far as I understood. And it's not only happening at the guard cell level, but also these kind of energy metabolism related signals might come from other parts of the leaf, like inside from the really photosynthesizing tissue. So I'm not sure if I explained fair enough, but it's let's say it's somewhat hot topic, but not really mine.
Thank you anyways, that's nice.
Vincent?
Do we have time? Okay. Hi, Yasmin. Thank you very much for a beautiful presentation and very surprising also. And thank you for the surprise for the KUVA Research Days to start with plant pathology. That's great. Yes, and so I don't have a scientific question. I have maybe more an aesthetic question, like to contextualize this in the KUVA Research Days. I'm really fascinated by the effort, not to say the interest you show in visualization and the care which is put in the data visualization. And I think this is a bridge to what we are going to do also later on, and to kind of like address epistemic / aesthetic registers of representation. So I'm just curious, what is your relationship to that? Do you think that putting effort in the visualization and aestheticizing, or like trying to bring aesthetic register representation, opens meanings to the or potential reading or another entrance to your work, or is it something else? And just as an experience, for example, you speak about stomata, so a mouth, and suddenly the mouth, which is like horizontal lips, become vertical lips in one, and vertical lips might refer to other orifice of the body, like more genital or more, and so suddenly my mind goes somewhere else. I have another stack of images that come to my mind, and I'm wondering if you're happy with that or not. So yeah.
Well, I don't think I think about kind of these things. So I might do something but it's not deliberate. When I'm doing like visual presentations, I think them to look nice at least attracts people, and to have like visual cues, it helps people to, at least not to doze off. That's one thing. I think many scientists kind of lack, I don't know if it's kind of like time management thing or prioritization, but when you go to scientific conferences or scientific lectures, there's really maybe not that much effort put into how people present the visuals. It's usually like super hard data. Okay, I have all these graphs, but what do they mean? And I think having some kind of like something that looks interesting, aesthetic or pretty or not, but as long as it looks interesting and captivating, I think it makes us interested. And that's, obviously makes things, at least to me, easier to understand and to follow. So I hope that somehow answered your question in a way.
Yeah, thank you, and I think we can keep on talking about that, because of course when it's contextualized within another field of where the reading is maybe different on the forms, on the shapes, on the colors, then it might stop what maybe Nina mentioned in the beginning, some opening eye, surprising meanings and senses and relationship. Thank you.
Yeah, exactly. Thanks.
Yeah, and stay tuned for 5 to 6, because then some of the things you've seen here will have been transformed into more strange forms. So please do come, because you'll recognize or not recognize it. Yasmin, kiitos paljon.
Thanks so much.
Thank you very
Kiitos
much.
paljon.
Yeah, and hopefully everybody can continue their conversations with you. And
I'd be happy to.
yes, just one thing. It's one thing that's not added to the Wayfinder of the tables. Your beautiful molds that you took is lying on a page of Rebecca on White Reed's, Rachel White Reed's house. So it's right on there. I thought so. Everybody that it's not noted on the A4. That's the Wayfinder, but it's there, and it's Yasmin's beautiful leaf molds that she's made.
I can't wait to look all the beautiful objects. So thanks.
Thank you. We'll make up for that. Our next performer, Hanna Kurell. Hanna Kurell is a doctoral researcher in the Dockmus Doctoral School of the Sibelius Academy in the University of Arts Helsinki. Her artistic research project focuses on gender performances of song recitals in the culture of Western art music through a new materialist lens. As an opera singer, her artist name is Hanna Rantala. During her career so far, she has performed over 30 leading and supporting roles of her voice type in the Finnish National Opera, Savonlinna Opera Festival, and Royal Danish Opera, among others. And without further ado,
Yes, good morning, and thank you, Yasmin, for a very, very fascinating presentation. This last question will lead nicely to my presentation. I will talk about breathing from more not a visual but more kind of experiential level, and of course plants have very much to do with human breathing, because we couldn't do it without plants, obviously. Great, thank you. So the object I have today is actually a piece of music composed by Haya Chernovin. Here's the front page of it. So, Adiantum capillus veneris, it's the Latin name of the southern maidenhair fern, and it is a plant that grows in temperate to warm climates and requires high-ish humidity. So often it grows on, for example, cliffs near water. It's also a common house plant. And it is cultivated for landscaping uses also. It has medicinal properties, and these are known in at least some Native American cultures and the traditional medicine of Iran. And it's used for treating, for example, rheumatism, jaundice, insect stings, coughs, and so on. So it's a quite a versatile plant. So, Chaya Chernovin composed 3 pieces for voice and breath called Adiantum capillus veneris 1, 2, and 3, with the subheading Etudes in Fragility for Voice and Breath. She composed them during 2015 and 16, during which she was also composing her opera Infinite Now. Her project has been to try to find ways of using the human voice as an instrument free of the connotations and meanings that are usually attached to it. So So when we hear someone speak or sing, we can usually deduce their age or gender, health, and in some language contexts, even their class. Also, singing technique or style of singing has its own connotations, like opera singing has different baggage from singing in folk music or rap or hymns or whatever. So Chernovin is interested in freeing the voice from these burdens or this baggage. So let's now hear what this piece sounds like. Thank you. So this is what the plant looks like. And my questions today are, so why did the composer choose this particular plant? Why not something, I don't know, big, fancy, rare, unusual, or something? And what's the meaning of voice and breath in conjunction with this plant? Why? Why did she compose it like this? I'll shortly show you some bits of the score, and you'll see it's very exact and precise. So why? I think these questions are connected to kind of a web of relationships in this piece, and also performance. Of course, this is a question of music ontology. What is, or where does the music live? Is it in the score or the performance? So the questions are connected to the relationship between the score and the performer. Of course, also composer, performer. Score is kind of a tool in between there. Then exact and free. And of course also human plant, since it's named like that. So to me, a score translates into a series of actions, or it's I think of a score of music as more like a set of instructions, or something that the composer has kind of pictured, but in that sense, the score isn't the music. When the actions are dictated very precisely, like here, you can see, it gives a very very different sort of embodied experience to more free scores or improvisation. So instead of something I can create out of my own body, out of kind of in my own terms, I have to kind of half force my body into a very rigid set of system that comes from outside of me. So here in this piece, you can see that the tempo markings are there, and for example, in the first and the second system there, you can see that, like, what is the difference between, like, there's bar 4, the second and the last beat. What is the difference between those? You can't really hear them, or you can't really, or of course you can hear the difference because the first one is hic and the second one is dl. But the rhythm, like, it's impossible to execute so that you can hear the difference because it's such a tiny, tiny difference. So what is the point of this? One might ask. So also inhales in this piece are dictated. So whenever the triangle there is facing that way, it's an inhale. So the piece begins with an inhaled, and the triangles other ways are then exhales. So usually composers don't decide when a singer inhales. I can decide it myself, and I can also make space for my inhalations with phrasing and so on. At least in the mainstream repertoire, I sing quite a lot. But in this score, the inhales are, or quite many of the inhales are notated, and even some of their lengths are noted. So like you see there, bar 90. There's it begins with an exhaled, and then on the next halfway through the next bar, it changes into a inhaled ah, and then you're at the same time you're supposed to then turn the ah into an e, and then change it back to an exhale. So yeah. It took me a while to learn this piece, but now I've performed it quite many times, so now it's not such a struggle anymore. So these are just some of the difficulties with this piece. Also, one aspect of using breathing sounds as texture in music is that breathing sounds are, of course, very quiet. So you usually need amplification, but also breathing sounds, something that I noticed during rehearsing this piece was that I started to hyperventilate. So I had to kind of adjust the dynamics, even though the dynamics are very quiet, but I couldn't kind of start doing very, very loud ones because I just started to hyperventilate. But the more quiet a sound you do, the more there is a risk that the sound actually breaks then and doesn't come out. So anyway, this is a very humbling piece to perform. And I wonder if that is actually the central substance of this piece. So by imposing this really strict frame of physical actions in conjunction to naming it after a plant, the piece is perhaps an attempt to remind us that our ability to breathe is dependent on these plants that we take for granted, really. And maybe to remind us that we are fragile and most definitely not the biblical king of all creation. So, for the rest of this slot, I think we could focus on page 11 of the score, which you see here. And let's do some of it together. yeah, I have chopped some of the elements here into a set of shorter exercises. So let's see how far we get. And just as a reminder, if you start feeling dizzy while doing this, stop what you're doing. Take care of yourself. But I'd like you to pay attention to while you do this. Where in your body do you feel them, and what do these exercises tell you of your breathing and your relationship to breathing? And yes, so feel free to stand up and move around while you do this. It might be easier and more relaxed that way. So let's start. So first we'll do exhales with different vowels on the count of 2, 4. So I will count 4 here, and we do an exhale. So let's start with just exhale, ah. And then e. And i. O. U. Good. This is the first element. Then we'll try some inhales. So these might make you cough. So I advise quite a relaxed inhale. So don't try to do like this, because that will make you cough. And we'll do slightly shorter inhales because it's more difficult. So I will count to 3. And let's start with again. No, breathe out first and then breathe in. And then relax, maybe swallow. And then breathe out again. And this time we do E. Good. And E. And O. And finally U. Good. Then we'll try to exhale a scale. So start from high and go low. So just like that at the count of 4. Let's see. There are many elements to this now, so this is slightly more difficult. So let's start with A again. Yeah. Now I start with E myself. Okay, let's try again. A. Good. And let's now do E, since I quite instinctively tried to do the same thing as there. So. As you can see, it's possible. And even clearer scale, it comes when you change from E to U while you do it. So let's try that. So that would be the same, kind of the same as in the second system there. So. See? And then let's try. There are different. Let's do a scale with so that we start with A and turn it into an O. So.
Good. You're quite good at this. What's our time? Yeah, we have still. Let's try inhaling a scale. So this one, let's do just a very small scale. Let's do so that we start with O. So exhale and yes, inhaling, but you have to start inhaling with an exhale. This is, for a singer, this is a very, very difficult thing, because always when I start singing, I started with an inhale, but if I inhale before I have to inhale, then I can't inhale anymore. So, but with inhales, it's peculiar, or it's quite difficult to do a scale down, because when you inhale, let's inhale with an O, for example. As you see, it almost naturally does a upward scale. So if you want to do a downward scale, it's more difficult. So let's try again a downward scale with O. So, and inhale. So the trick to do it is to change the shape of your mouth. So while you inhale, you have to open your mouth a bit, so that gives it a bit more space to resonate or whatever. Yeah, I think we could stop here, because otherwise we'll run out of time, but I can answer a few questions if you have any. Thank you.
Kiitos, Hannah. So I'm just going to open it up to questions, so we can take a few if there's any in the room.
And thank you so much for such a fascinating talk. I have no background in opera singing or music or stuff like this, and for me this seems very, unreal that this kind of music exists. So could you comment on a few notes on what is it like to work with material that seems so, maybe not controversial, but so out of the box when it comes to what a layman thinks, for example, music is, or opera singing is, or classical music is. So could you comment a few words on that?
Well, for me, it's really nice, because I've spent the last 15 years singing opera, and it's one way of using the voice, which I rather like, but it's quite restricted in that way. So for me, it's really kind of refreshing and freeing to do stuff like this, where I can then use my voice in different ways, and also quite surprising, because like I said, opera singing has its own baggage and meanings and blah blah blah. So this then kind of doesn't, in that sense. Of course, contemporary music is a marginal thing, like perhaps all contemporary art, but within the music scene, contemporary music is it's in the margins. But I think it's important to do this kind of stuff, because otherwise we'll just end up being a kind of live performance museum rather than live art form that has the potential to develop into something and perhaps be in contact with the current society and issues.
Thank you so much. That was so fun. During your rehearsal process, did you develop some sort of relationship with the Maiden's Hair Fern? Did you have them near you, or chew on their leaves, or something like that?
No, I have to admit, no, I didn't. I will also admit that I am a very non-visual person. If you were to visit my home, you would see it. But I somehow the biggest question for me with this piece was like, what's the connection with the plant? Like, why did she name it after a plant instead of, I don't know, just breath or something more? So this presentation was kind of my attempt to understand the connection between the two. So, yeah. But that's an interesting thought that I could actually do it. I've understood that the maidenhair fern, even if it's common houseplant, but it requires quite a lot of water and humidity and care, which when it comes to plant, I am not capable of. I've in my life managed to kill a cactus by not watering it, so houseplants are not safe with me.
I have a question concerning the notation. You emphasize the precision of it, and my question concerns the phonetic layer of it. Is there some kind of universal phonetics behind or in this kind of scores? Because obviously in different languages, the phonetical universe or the relations between the phonemes are different. So is there some instruction, or is English, for example, the calibration here, or?
Yeah, it's there is such a thing as International Phonetic Alphabet. It's not used here though, but I think it says here there's 2 pages of performance instructions at the beginning of the score. So basically explanations of what the different notes mean, and also some words on amplification and so on. Yeah, it might be that it says in the first part that these are Italian vowels. So, but yeah, usually composers define what they mean if they use notation like this and not the International Phonetic Alphabet. So yes, that's a pertinent question, because of course, if that would be English, then the would be, and the eyes would be, or the long would be something like that. So yeah, yep. But it's a question of notation and instructions. Yeah.
Thank you very much also for making us try it, as I think, which is precious also with artistic research, when we can also experience it with you. I spotted the date of that piece is 2015, and I was thinking it's also like the moment where the Anthropocene concern is booming in the art, and especially in performing arts, where there's this process of kind of reversing the mimesis dynamic. Is not anymore to anthropomorph the non-human, like singing like a bird, but it's kind of humbly to bring the human tools and capacity in trying to approach the non-human being, agency, state. I'm thinking, for example, of Esakit Kopelto and other spaces group who try to embody a deer or a wolf, like in Deer Safari, or how to be a gastropod. So, we're not in theater doing anymore the flower, but we try to understand how a flower can come through the body. And I think like Maja Edvanen is working with mushroom and choreography. I think there's in scores that might be also I don't know, connected with that. I don't know if you want to say something about this, but yes, I'm just wondering if that piece is part of, in music, because I'm ignorant in music, is part of a movement linked to this Anthropocene boom, where we are starting to kind of like really consider how to sing vegetal, mineral, animals differently.
Yeah, it's one possible option. I am not aware that Chernovin herself kind of deliberately did that, because these 3 pieces are kind of etudes for her in how to use the voice in this manner. Because in her opera Infinite Now, there's quite a lot of this sort of voice use. So she basically did this as a kind of an exercise in how to use this. The first part of these pieces was composed for the NDR singing competition in Germany. But that is, of course, one possible explanation for the vegetable or plant or vegetation connection. Also, Haya Chernovin is from Israel. She has lived in Germany and USA for quite a long time. But I reckon this plant is something that she grew up surrounded by.
Great.
I might have more a comment than a question, but while you were performing, I'm not that much into music, but I was imagining how the score would be, like how, it would be notated. And I was having as reference, for example, these John Cage notations, because having been in Maria Lang concerts, and, she's quite pedagogical, being taught by Cage himself. And then it was very surprising to see the scores, like so precise, and how extremely should be performed, not being faster or slower, and it's very much underlined. I don't have that much to say, but it was quite a contrast also coming from the visuals and the images of the microscope, and being inspired by Yasmin's presentations. We got, all these visual images, and then suddenly something so beautiful also and so inspiring comes from such a precise notation system.
Yeah, here that's a good point. Like John Cage being the exact opposite to this, with his graphic notations and so on. I think this score is kind of a performative text in that sense that it performs its preciseness. Just looking at it's there's a meaning to it being notated so precisely that it's impossible to execute. Like a slightly more precise execution would be that I would have a click track in an earbud, so I could do the tempo changes more precisely. And actually, I did that with the third piece, which has 2 recorded tracks, and then I sing live to the third one. So I recorded the two tracks with a click track, so that I could synchronize them. But yeah, it's also the click track is one really kind of it's a control aspect as well, and it takes away even more of the freedom in that sense.
Kiitos, Hanna. That was just the most enjoyable wake me up to so many things session, and thank you to the audience for such sensitive observations and questions. I keep forgetting that the YouTubers are able to, I should announce this, YouTubers watching can actually pose questions, and we will field them in the room itself. So sorry for everybody at home that wanted to ask questions. Hopefully you can track the 2 speakers down and email them the questions, but apologies. Okay, so now the curator steps in again, and she's going to do a 10-minute little interval. And what these are, they are referencing quite a few objects that I was not able to source and actually bring into the space for you. Many of them are the ones that are sitting in the Tulla Museum Hospital or Tula Hospital Museum. And those things are not in that space anymore. They're now in storage in Vanta. So there's images of them, which is not the same. But I'm still using the images as material for you to evoke just the possible connections between these. Ooh, no, sorry about that. So it's just the interval number 1, that one. Okay, and then brilliant, presenter view, perfect. Okay, so I'll be sitting here and, okay, short interval. Objects: anesthesia workstation, X-ray goggles from the Tula Museum, an equation representing respiratory difficulties in plants, and infrared drone footage of trees affected by bark beetle infestation. Becoming part of an academic community requires a series of encounters with the objects involved in the practice, the research materials, tools, furniture, text, and symbols, and the categories that apply to all of these. As individuals' familiarity with these deepen over time, through the communal activities of attending lectures, reading disciplinary literature, and learning and discussing current ideas in the field, participants become insiders naturalized to their community's biases, habits, norms, and conventions. While these processes enable specialized knowledge production, they can also have negative repercussions that hinder discovery, as illustrated by historian of science Thomas Kuhn's well-known discussion of the development of X-rays. In 1895, Wilhelm Röntgen, professor of physics in Würzburg, Bavaria, accidentally discovered X-rays while testing whether cathode rays could pass through glass. Using a cathode tube covered in heavy black paper, Röntgen was surprised when luminescent green light escaped and was projected onto a nearby fluorescent screen. Upon further experimentation, he discovered that this mysterious light passed through most materials but left a shadow of solid objects. This discovery was met with shock and disbelief. Though X-rays had not been ruled out by established theory of the period, which accepted radiation such as visible, infrared, and ultraviolet, they violated the expectations implicit in the design and interpretation of established laboratory procedures. The decision to employ a particular piece of apparatus and use it to a particular end carried with it the assumption that only certain sorts of circumstances will arise. In acknowledging that this equipment, widely deployed since the 1890s already, produced these rays, was also to concede that their existence had gone unnoticed for quite some time. In 2018, whilst in conversation with Dr. Jane Yates, the pathology collection curator at the University of Cape Town, she revealed that most of the plants she had introduced to the venue of the collection had mysteriously died. The venue consisted of 3 interconnecting rooms filled with 3,500 diseased human anatomical parts, contained in formaldehyde, categorized by organ, then disease type. In response to what Dr. Yates shared with me, I X-rayed a selection of indigenous plant specimens, known for their local healing properties, the starting point of many diagnoses in human medicine. There should be sound here. Sorry. Okay, there he is. In one of the chapters of the 1959 publication, Plant Pathology, an advanced treatise, two scientists, Ikuzu Uritani and Takashi Akazawa, formulated an equation for depicting what a change in the metabolic process of respiration in a plant might look like. Using a cathode tube covered in heavy black paper, Röntgen was surprised when luminescent green light escaped and was projected onto a nearby fluorescent screen. Yulei and Marina introduced the video of breathing in, breathing out as follows. We are kneeling face to face, pressing our mouths together. Our noses are blocked with cigarette filters. Yulei says, I'm breathing in oxygen, I'm breathing out carbon dioxide. Marina, I'm breathing in carbon dioxide, I'm breathing out carbon dioxide. Yulei, I'm breathing in carbon dioxide, I'm breathing out carbon dioxide. On a more personal elegiac level, Vanishing was created when Lawrence's father was on life support in hospital. An anesthesia workstation from the Turler Museum. In conversation with University of Helsinki researcher Evgeny Lupatin, he shared this image with me. Drone infrared footage of spruce trees showing the trees that are reflecting sunlight due to a bark beetle infestation that affect their respiratory cycle. A moment of observation while visiting the Wiki Plant Sciences Center, Helsinki. Like ancient seafarers, dung beetles can navigate using the starry sky and the glow from the Milky Way. After locating a fresh pile of feces, dung beetles will often collect and roll away a large piece of spherical dung, then climb on their dung balls and dance around in circles before taking off. This dance is not one of joy, however. The insects are watching the sky to get their bearings.
Blue moon, you saw me standing alone, without a dream in my heart, without a love of my own. Blue moon, you knew just what I was there for. You heard me saying a prayer for someone
Kitos. That was the first interval. Okay, so you've still got to look at me at this stage, and I'm moving into the mystery objects part of or the magic objects part of the presentation. Yes, thanks, Pavel. Okay, so just to give you a little bit of background on this, this is not my presentation. This is a compilation of people that I've gone to see and questioned in terms of two objects that I found online on the Fina Museum digital repository of objects. And it's two beautiful little birch balls. And you don't get a sense of scale as you look at these things online. And when you look at them online, they seem much more, they seem much larger, like a lot of things do when you look at things online. You don't have that sense of scale. And it was two little birch balls that were located in the Satakunta Museum in Pori. And I contacted it was in Finnish, all the information, and I contacted the curator of the museum to start kind of finding out what these objects were, because they were termed, when I translated it, as magic objects used in healing rituals. And that was the only thing that was actually, that was part of the record online. When I contacted Johanna, who's the curator of the museum, she said that there's unfortunately scant information in even their museum records. The provenance is scant. There's almost nothing there. They just basically shared the information that they did have on the online space as well. So they were in some way obscure objects that are in one of the displays in the museum. The display features various forms of healing and magic, and of course, those terms like magic are very problematic in the current day, because it flattens quite a complex constellation of ideas and historical practices and very specific practices. So my impetus was then to try and see if I can connect with as many people as possible to find out more about these objects. So it's the two little birch balls, but it's also a little birch ring that also sits with these in the same display case. And I'm going to delve into this kind of trajectory that I started for finding out more about this. And we'll have our coffee break after this. So. Two small birch balls and a birch ring have been sourced from the Satakunta Museum in Pori, where they form part of the permanent exhibition display, Signs of Life. Titled Magic Objects and classified in the museum cataloguing system as woodwork, magic and sorcery, folk belief, and luck and chance. These objects were used in healing practices. Like many objects classified as magical in museums worldwide, scant or no information is available on their local use and their history. This session documents the ongoing quest to find out more about them and their provenance. The color of the object is brown. There's sound on this one as well.
Yeah, I think this looks like the disease or the. Do you know the fungi that lives in birch that causes this?
Like the abscesses.
Yeah, it looks like that.
I think it's Agrobacterium, you know. It causes, yeah, because it's callousing. It's just bulging up like undifferentiated tumorous growth.
Okay.
So maybe it's like one of these bowls that like they use them to make the
Kuksa.
Kuksa, yeah.
Oh, okay.
I think it's the same thing.
Is it Agrobacterium?
I think it is.
Oh, okay. I don't know.
I think, don't quote
Okay.
me, but
Yeah, okay.
yeah, that's
So-
the response.
A disease is causing the growth to occur.
Yeah, it's like a tumor in birch, and
Okay.
there's like a. I think they make some tea out of it. This this superfood tea. I don't know. I can't remember the name right now. Pakuri tea.
Oh.
Okay, yeah, that's in the Finnish term.
Okay.
And it's supposed to have a lot of antioxidants, and yeah, it's supposed to heal you from your
Okay.
diseases. But yeah, this is just I think this is just a small ring, and usually what you see is like a big ball, like a tumor-like tissue.
Because my feeling was that it was implemented by, you know how people encourage a type of growth pattern by doing certain things, like you're doing with your compression ones. So, but you're thinking this is just a natural occurring thing because of the disease, and it was then kind of isolated by the people who used it for various other reasons. It was something that looked unusual, it was isolated, and then
Yeah, that's what I think, but of course I don't know. It's difficult to say, like when it's sort of isolated and it's a couple of 100 years old or something.
But it's like petrified as well. And would it be used in the same way as like a bracelet, as something for a child?
see, the information is very scarce on what it is in the museum records, actually, except that it's titled under magic objects and kind of healing kind of objects of sorts.
Okay.
So
So-
like shamanistic almost.
Yeah.
But if you look at the wood inside, it doesn't look like wood. It's something sort of. It's really weird.
It's
Or is it?
the layers are so different. I mean, the outside obviously that's become from the outer part of this tree, and the rest has been sort of the wood's there. So whether it was a part of if it was part of the bowl, or if it was something that was just taken from a branch, but you know, because the fibers are so, it's in its natural shape for sure.
Huh.
Too much wood.
Yes. Yes, so I don't know where to begin with the folk or with the magic or or maybe it's easier to start with the late 19th century, because that's when these objects were started to become collected by different kind of like school teachers and students and this kind of enthusiasts who were interested in recording the history of different rural areas in Finland. And it was thought that there was this kind of folk culture that was disappearing at the time, and they wanted to record that. And it was kind of like, at the same time, this folk culture was seen as being kind of outdated and belonging in the past, but also in the same time it was seen like something very like purely Finnish, something like that that could be used in the nation building of Finland, which wasn't even a independent nation at the time yet. So it was in the future. Yeah, but the folk, the idea of the folk was rural people who were not highly educated, and of course there's always problems with this kind of terms and concepts that always limit themselves, and of course, I mean, there are different kind of people in a rural setting as well. So where do we find the limits of the folk? And of course, in towns also. But the collection work was not done in towns because it was thought that the real folk culture was to be found in the rural areas.
Mm-hmm.
And people still remembered and some still used magic objects in their everyday life at the time. And they were also talking about these magic practices to the collectors, and sometimes also these objects came to the collectors, usually when the objects have been used by a grandparent and the grandchildren were not anymore kind of interested in continuing using them. So then they ended up in the museums. And the magic practices, of course, they were something that was part of the everyday life. Many people kind of knew how to use these kind of objects, and they were like part of the tradition of how people lived. And especially in healing practices, when someone was sick, so they kind of knew how to heal different kind of common ailments. But also when the livelihood, they were working in the fields and taking care of cattle and fishing and doing forest work, there was always this kind of dangerous stages in the work where everything could go wrong. There could be bad weather, or animals could get sick, or fish could maybe disappear from the lakes, and it was important then that these kind of things did not happen. So it was different kind of practices that people did, so that they could make sure that everything goes well and smoothly, because of course it was a question of life and death.
Mm-hmm.
And yeah, so what magic is, of course, it's defined from the researcher's perspective, and usually it is kind of seen like something that is not rational, something that is kind of extra in some way, and also unnecessary.
Mm-hmm.
But for the people who used them, their worldview was different, and it was very important that these things were taken care of and done in this way, because for example, the worldview included different kind of guardian spirits in nature and in buildings, and for example, the guardian spirits in nature, if you did something wrong when you were walking in the forest, maybe kicked a stone or something, then you might anger the guardian spirit and get sick.
Mm-hmm.
So it was very important to know how to behave, and also if you accidentally did something wrong, then you should also know how to deal with the situation so that you could get better. So it's a very different worldview than the one that we are maybe seeing today. So I kind of define magic as practices that also include the kind of way of thinking, that there is complex relationships between things that like seemingly things that are seemingly separate are actually connected in complex ways, and there was some kind of symbolic connections between things, and you could manipulate one thing and then have a effect on another thing because of these connections. So that's the way I usually define magic, but there are many kinds of definitions, of course, and the term is problematic also in many ways, because it kind of carries with it these kind of meanings of something that is not true and not rational and at least unnecessary, if not something that is to be frowned upon.
Kate, I didn't know if you were going to be here, and I don't want to put you on the spot. You can just do this with your head if you really don't want to maybe introduce your own project, because it feels violent for me in this instance to sit here with you here that can potentially speak to your own kind of response to this. I don't know. No, just it really. What makes you feel most comfortable? Should I just present it? Yeah. Okay, brilliant. No, all good. Okay, so I realized really late that Masters in Sculpture student Kate Ruck's practice actually really intersects with these objects. And the museum practices that surround these objects. And I sent them to her, and she responded back saying that she will do the following projects. So the project explores the concept of magic objects within our diverse community, aiming to highlight the contrast between academic ideas and lived experiences. To contribute, POC were asked to describe 2 magical objects or a complex single object. And these descriptions were fed into an AI for visual representation. The resulting seeds clearly establish correlations between the contemporary functionalities of black box AI systems and their congruence with the archival systems employed in museums. Academia can function as a hegemonic narrative if those responsible are not discerning. In the absence of intimate and human knowledge, and in the exclusive presence of empirical scientific facts, what configuration of historical discourse remains?
When I was doing this project, I was also thinking about 3 different important things, and kind of defining what magic is as a term, thinking about the historical perspectives, and specifically about indigenous magic. So I think magic as a term has always been relegated in general to the realms of fantasy and dismissed as superstition when looking at it within the Western academic discourse. But if you're looking at the framework of indigenous studies, magic takes on a more nuanced and culturally embedded significance. And this is because indigenous cultures around the world are practicing traditions that intersect with spiritual practices, and there's also animism and a more holistic understanding of the interconnectedness of all living things. Also, if you're basing your idea of magic on historical perspectives, I think the colonial encounter between Western powers and indigenous societies has always resulted in the imposition of Eurocentric worldviews, and it's often dismissing the indigenous knowledge systems as primitive or irrational. Magic practices are deeply ingrained in indigenous cosmologies, and because of that, they were either criminalized or suppressed, and that also contributed to the erasure of cultural autonomy. If you examine historical accounts, it kind of allows us to understand the impact of colonization on indigenous magical traditions and the need for this reclamation. I think the last thing to recognize is that indigenous magic is not a monolithic concept, so its manifestations are as diverse as the cultures that practice it, from healing ceremonies to shamanic rituals, and also the acknowledgement of sacred landscapes. I think magic within indigenous studies encompasses a really broad spectrum of practices. They always seem to reflect, though, the profound connection to the natural world. I think if we take the time to understand these diverse expressions and how they're challenging the Western dichotomies of natural and supernatural, it would result in a more inclusive and respectful dialogue.
Some thoughts on the Anarchive, a term coined by researcher Corinne Zyman, an artist, curator and scholar committed to critical engagement with colonial archives and collections, specifically those holding strands of Kwekwe pasts. Zyman is currently a researcher and research coordinator at the Research Center for Material Culture.
Where is the past? We carry personal memories in our minds and bodies, but we also carry stories of occurrences at which we were not present, even from times that precede our births, that we know through what others have told us. In European colloquy, there exists a tendency to imagine the past as being behind us as we look forward to the future. And yet we continuously narrate the stories that we carry mentally and bodily, thereby inserting them in the present. One can thus argue that the past is not really behind us, but with us, constantly imagined and reimagined in public discourse through historical narrations. However, the creating of these historical narratives necessarily provokes a question of sources. What kind of material is apposite to serve as the foundation for the stories that we tell of the past? Stories that need to account for the wounds we, particularly those of us living in places deeply disturbed by European colonialism, are suffering in the present, but that should also help chart the futures we want. Seeking to loosen the ossifying grip archival materials has habitually held on historical narration, I wanted to find discursive and methodological means to reveal the extent of absence in archives, particularly those fashioned along the lines of colonial epistemology and in its service. The consequence of the centralization of absence is that archives are shown to be proportionally trivial, and if perceived in this relationship of scale, their capacity to control what can be known and narrated about the past is rendered absurd. My frustration with the incapacity of colonial archives to answer the needs of a post-colonial and post-apartheid society led me to focus on the troublesome archival silences themselves and grapple with their implications. To this end, I conceived the Anarchive as a sign denoting everything that is absent from archives, understood in a profoundly expanded sense. The anarchive refers to everything that cannot, by definition, be contained in archival documents, including the full sensorial experience of life, its non-linear temporality, unrealized potentials, and immaterial networks of intersubjectivity. Whereas archives as a medium can be said to filter out much of the intangible experience of lived life, the anarchive is contrariwise defined by them. Being founded in the intangible, the anarchive brings into view the vastness of absence in archives, since such absences refer not merely to that which might have been recorded but incidentally wasn't, or that which was recorded but subsequently lost. Rather, the anarchive is constituted by that which in the first place escapes the documentary matrices that make archive possible. What is more, the anarchive locates custodianship in the past, not in archives, but in various other intangible forms. Consequently, an anarchival methodology to making history, whether in formal or informal ways, in written, spoken or performative modes, eschews exclusive reliance on slivers of archival material as evidence. Even though it reframes the relationship between archive and evidence, the anarchive does not suggest archives have no value. Rather, it locates that value in the imprints of absence that archives hold, the ways in which they can help bring into view what is not there. From an anarchival standpoint, archives are invaluable not so much for preserving strands of the past, but rather for making visible the imprints of absence, of that which they failed to contain. I contend that this is the future productivity of archives, to enable us to see the imprints of absences within them. Even though all archives are haunted by the anarchive, certain colonial archives, often those that are vast and rich in detail, are particularly well placed to hold imprints of the presences that are absent from them, and are consequently more generative of work that makes the anarchive perceptible, sensible. Whereas historiography, as developed and exported from Europe, has instrumentalized archives as constituting the limit of what is considered evidence, the anarchive makes us aware that there are many other sources we might draw on as we narrate the past. Thus, the anarchive offers a constellation of archive, in which the written colonial document can no longer act as the despot of historical narration, especially where it concerns the histories of people relegated by colonialism. Instead, the anarchive privileges other practices whereby we perform our custodianship of the past, practices that keep alive memories and ways of knowing in defiance of coloniality.
And that's the end of the first morning session. I would really invite you to maybe look at the tables during the session and have a look at the table, enjoy your coffee, and we'll be back here at 11:20 for Nika Johansson's presentation. Kiitos for your patience.
don't know. I mean, because I will...
Okei. Miten sä aloitat sillä tavalla? Aloitatko täältä?
Nämä on varmaan koko aika suurin piirtein.
Se on koko ajan tuolla.
Joo.
Mut jos niinku se, että mitäs niin jos basically how much are you pointing at the object?
Well, my initial plan was to just mention some of the specimens that we have over, so all of it should actually happen there. So for online people, I don't need to worry about this.
I think we're going to maybe try to just at the right at the end make a plan of the changes.
I mean, I will be planning table. I can do it continuously over the presentation. So then it's gonna hear every now and then. Just we need to keep lights on. We can also make it so that lights goes on in the middle, or like so we start like that. Yeah. Yeah. Yes. Yes.
Jos olisi ollut enemmän aikaa, niin mä olisin pistänyt teidät kaikki keskitään.
Are you happy?
Joo, ja siis voi olla itse säveltäjä.
Like reverse multitasker. Definitely bad idea.
Hänellä on joku. En oo kysynyt häneltä.
Ehkä sen mielestä se kasvaa uudestaan.
Niin, se voi hyvin olla. Se voi hyvin olla, että se on yksinkertainen asia, mutta tietysti näin kun esiintyjänä teen siitä vaikeampaa.
Tässä on varmaan paljon enemmän niin kuin muutakin.
Or shall we just skip it and play the video?
Yeah, it's not even on video, but
Okay, we just skip it.
yeah, you're gonna have to do that. I'm sorry.
Okay, no, don't you be so great.
It's very sad. I'm gonna say it's going to go up on the wall afterwards.
Okay, right. Okay. So, yes, it's about time to continue.
Yes. Oh, I'm taking this. Where's my notes? Okay. I hope you feel replenished with your caffeine and sugar. So the next session until 1 o'clock starts with Nico Johansson. Nico Johansson is a biologist and doctoral researcher at the Finnish Museum of Natural History and University of Helsinki. He is interested in all things fungal, algal, botanical and more. Currently working with dispersal interactions between birds and fungi. Using classical collection-based research together with emerging environmental DNA studies, he tries to make sense of the diversity of life forms and interactions in the natural world, and to provide data and inference on how we can conserve biodiversity in the era of global biological change. Niku, tervetuloa.
Thank you. Okay, perfect. And the microphone is on. Perfect. Hello. Welcome. Thanks for coming, and thanks for the invitation and the introduction, Nina. It's fantastic to be here. My name is Nico Johansson. I'm a doctoral researcher, biologist, ecologist. I mostly work with plant sciences, fungal sciences, diversity of fungi, algae, lichens, and all things around there. And I come from a slightly different point of view to biological sciences than Jasmin did in the morning. So I work with larger things. I also use smaller molecular materials to study those. But my research topics are on the ecological scales, looking at how nature works, how species are interacting on the macro scale as well. And my thought behind this presentation was first to give you a little bit of a glimpse of what's happening in Natural History Museum collections. So I know that in the audience, most of you have a background in humanities and arts and art research. So I thought that I will give you a brief introduction of what the Natural History Museum scene is really doing right now. What do we actually have, and how do we research on these objects that we have in our collections? And then later on, in the latter part of my talk, I will give you an example of collection-based research in my own PhD studies, looking at these objects and how we do ecological studies with them. So hopefully after this presentation, you will have a bit of a better view of what the scene is like in the Natural History Museum world and how these collections are maintained and why do we have them. And classically, we tend to think that natural history museums are kind of these exhibitions of biological wonders around us, and having this kind of almost archaic idea of showing the poorly educated Finnish people what a lion looks like in a public exhibition, and showing the wonders of the natural world around us. This is actually a real picture from the Natural History Museum in Kamppi in Helsinki from the 1920s. So a part of that, of course, looks a bit of this cabinet of curiosities. But the actual history of Natural History Museum collections goes way beyond that. And it pretty much all started with plants and the practice of scientifically collecting plants interties with herbal medicine all the way from historical times. Humanity has used herbal medicine pretty much all of our existence, but in the 1400s, 1500s, 1600s, we started to systematically collect botanical specimens. Just for species identification purposes. It's very important for whoever uses herbal medicine to know what kills you, what doesn't kill you, what's good for you, what's good to eat. And then in an era where books and scientific studies are only just developing, pressed herbarium specimens became a way to communicate ideas in a more systematical manner of how plant diversity can be of use for us. I work mostly with lichens these days, so I had to have a lichen example here as well. But the collection of economically and medicinally important plants created this kind of idea of having institutionalized herbaria. And then later on in the 1700s and onwards with Carl von Linné and all of these people who started to look at the biodiversity around us in a more systematical manner, initiated the study of actually studying the diversity of, say, plants, animals around us. And a key part of this was also to collect specimens that we can objectively study, especially in a systematical manner, trying to identify and figure out what is the flora of a country like Finland. What species are there, how they differ from each other. This required the collection of specimens for detailed study. And these collections became the foundations of the large Natural History Museum collections that we have these days. And this is a cumulative process, of course, so the specimens keep on coming. And nowadays, these modern natural history museums are large specimen banks of this historical stuff, but then also new stuff coming in. So I decided not to tell you what, in conceptual terms, what the modern Natural History Museum collection looks like. I will use us as an example. So Luomus is the Natural History Museum, Finnish Museum of Natural History, hosted by the University of Helsinki. It's Finland's national collection, so it's all owned by all Finnish people. And we are a quite a good example of a medium to large size natural history museum in the world. We house about 13.5 million specimens and objects in our collections, plus a backlog. Of course, we haven't had time to incorporate everything, so maybe 15 or something million specimens. And classically, natural history museum institutions are roughly divided with zoological departments and botanical departments and geological departments, like we are as well. And I will now give you a quick run through of what material is among these 13 million specimens, and give you ideas and inspiration what the collections actually look like, and then I'll tell you a little bit what we do with the collections. So this is an overview of our zoological department, about 10 million specimens. Most of them is insects, as you can see here. You can wonder if we had about 13.5 million specimens, most of them, the vast majority, are insects. You can think of why. One of the reasons, of course, is that insect diversity is very massive, so most of the animal diversity actual in the world is insects, so there's plenty of them to collect, but then also they're small. So most of these are pinned insect specimens, like here you see in the butterfly collections, so they don't take that much space, which is also convenient. We also have other invertebrates, quite a big collection. This includes mostly spiders, mollusks, worms, things like this. And a common feature for many of these things is that they're soft-bodied, so they don't really preserve that well dry as pinned insects. So hence many of them are in these curious-looking preservative liquids to keep the body shape of the animal in a way that we can use it for scientific study. And then, of course, we have a vast vertebrate collection as well, a bit smaller, and this is again also logistically reasons, because many animals, mammals for example, are quite large, so they take a lot of space. This is not just stuffed animals, so I will try to change you with the misconception that natural history museums are just collections of stuffed animals. These are skeletal samples, skins, bird skins like here, different kinds of ways of how animals, experimental animals, can be used and preserved for scientific research. And this is where I work with on the botanical side. Most of the botanical collections are of vascular plants, and most of them are herbarium-pressed specimens. You have an example here on the table. In Helsinki especially, however, the botanical side is very much known for their collections in non-vascular plants. So we look at bryophytes, these are mosses, liverworts, things like this, lichens, lichenized fungi, non-lichenized fungi, all different kinds of mushrooms and so on and so on, and then algae as well. And these are among the most important collections in the world. So the University of Helsinki Natural History Museum collections are probably in the most top 25-ish sized and importance collections in the world in general terms, but with lichens, fungi, bryophytes, we are among the most important collections in the world. So we are very visited, and many people who are studying the life history and evolutionary history of bryophytes and lichens frequently visit Helsinki because our collections are so unique and important here. And then on the geological side, a bit smaller, we have a collection of mineral samples, rocks and sediment, stuff like this, and then also meteorites and other geological formations, and then fossils, etc., from classical fossils to amber fossils to subfossils, for example, from Ice Age. We have a huge collection of mammoth stuff and things like this. The University of Helsinki and the collections that we have here, due to the geological history of Finland is not really rich on fossils, so we don't have a long tradition of dinosaur biology like many people tend to think that natural history museums usually do. But we also have something, of course, and do research on this stuff. And then we should not forget that many natural history museums, us included, also have a living collection. So we host 2 botanical gardens, one in Kaisaniemi and one in Kumpula. And these botanical gardens are actually part of the national collection. So the plants in there are actually living museum objects. They are specimens for scientific study and of course for recreation as well. So the tropical collections inside the Kaisaniemi greenhouses, there's a systematical collection of plant strains outside in Kaisaniemi, and then in Kumpula also we have this plant biogeographical point of view of looking at plants originating from different parts of the world and understanding that diversity in spatial occurrence around the world. And ethnobotanical collections also in Kumpula, looking at ornamental history of plant use, ornamental plants, medicinal plants, varieties traditionally used in cultivation, and so on and so on. Preserving these kinds of plants and the way that the plants are being used is also an important part of our work. So I always tell that when the gardeners are working and maintaining the gardens, they have this dual role that of course they're gardeners, they work to make the place look pretty and so on, but they are actually working as a bit of like a museum curator. So they are curating and managing the collections, even though some of the collections, of course, are live organisms. So it creates some sort of kind of interesting philosophical ways of putting what museum objects really are. And then finally, and not least, public outreach. So of course we have huge museums and the botanical gardens. They're open for the public. You can buy a ticket and get educated. We also do lots of other public outreach activities, scientific outreach, and this is really important and central actually for many of the stuff that we do. But here lies a bit of a misconception oftentimes that the Natural History Museum or Finnish Museum of Natural History is much more than just the public exhibition, and this is usually true for most of the natural history museums around the world. So you can't really equate the entirety of the museum into the public exhibition, although it's a central part of it. So we act more like a research institute that has a public outreach program instead of a public outreach program that has a research activity within. They are intimately linked. And on top of that, we are not only just a collection of objects, but also the expertise that we have. We employ about 150 people. Most of these are collection management staff who are actually day-to-day taking care of these collections, be they gardeners, be they botanical people, be they insect people. And we are maintaining these collections. It's our legal duty to take care of them. And with this, we have created a group of information and knowledge and wisdom about these collections and about the taxa, the animals, the plants that we work with. So we are also like a center of expertise and knowledge about this stuff. So the Finnish Museum's Natural History is the kind of official, almost official person who is, or entity who is looking after what is growing on in this country, for example. We are responsible to know what plants grow in Finland, what animals are found in Finland, and how should we look at them. After this brief overview, I'm going to introduce to a few topics, concepts, trends in the Natural History Museum scene that are really hot right now. So you can kind of appreciate or think about the way that these collections can be used in a more comprehensive manner nowadays in the future. And one of the central things that we think about in this scene right now is the concept of this the extended specimen. So oftentimes when we talk about specimen numbers or managements and so on, we think about the objects just as physical objects. But of course, if you think about the view of a scientist or somebody who could use this object for research, there is so much more that information from that individual specimen than just the physical specimen itself. So this concept highlights the fact that we have this overlapping, extending rays of information that we can extract from physical objects, be it just, of course, the physical object itself, but also a digital record of it in a collection management system, and then all of the label information, of course, where did it come from, who collected it and why, and so on and so on. But then it doesn't stop there. It doesn't stop what's clearly visible with the actual specimen. You have information about field notes of how it was collected, and maybe a genetic molecular sample of it, a tissue sample, information regarding the morphology of the specimen, this kind of stuff. And then further on, information regarding the taxon or the animal or the organism or the context of the specimen. Where does it sit in the evolutionary tree of life? Is it protected, threatened? Does it have, what's its world distribution, so on and so on. So you can imagine this kind of level of information that is extractable from a single physical object. And this is really a central point of view looking at specimens. So the information that we can extract from these specimens is actually quite large. And how to store this information and make this information available for scientific study, for artistic study, and so on is a key challenge on how do we communicate all of this information that is available from these specimens. Another kind of hot topic right now and a cool thing is that an example of the concept of this extended specimen, that nowadays we can really use these old museum specimens in innovative, modern molecular ways, for example. So this is an emerging field called museomics, which uses molecular studies and molecular techniques that we've heard from Yasmin, for example, on museum specimens. And this opens up research questions from very, very different parts of the that were really previously inaccessible. So we tend to think that for DNA work, molecular work, we really need fresh, high-quality material. But nowadays, the methodological approach advances in this field have been so vast that we can actually use fragmented old DNA from historical specimens in completely new way. And this unlocks things like this. These are examples from our museum. Peter Boksai, a curator for the vascular plant team, for example, have been looking at this ambrosia. It's a weed, it's an invasive weed. That is a huge problem in agriculture, and they sequenced genomes to look at the invasion history. But they used material that was collected in the herbarium before the plant became invasive. So you can go back in time to collect material, so already collected material that you can see, oh, what did the plant genome look like before it became invasive? And did something happen? Was something causing this invasion while it was invading? What happened to the genetic architecture of the plant for us to better understand how invasive species and biological invasions, for example, happen? So this is now just, we can't go back in time really, but we can go back in time with specimens that we have in the thing. And the other one here is a nice example as well. This is a moth collected from Madagascar. There are 2 known individuals of this species in the whole world. They're in Paris, and they were collected back in the day. And then it has become this kind of mystery in the moth research community of what the species is, where did it come from, and in evolutionary terms, does it fit in the tree of life anywhere? We didn't really know what it was until Victoria from our museum managed to extract DNA from these old specimens, and now we can place them in the tree of life of butterflies. And now we understand just another piece of the puzzle of the diversity of animals that we see around us. This would have been impossible without using historical museum collections in contemporary ways. And then finally, a huge thing in the museum scene right now is something called digitalization, digitizing specimens, databasing. So most of our collections, they used to not be in an Excel. So people tend to think that, oh, we can just go into an Excel and see what we have, how many specimens, where they were collected. Most of our specimens were collected before Excel was invented. Most of our specimens were collected before computers were invented. So actually, we have a huge issue now, and a huge task to do is to catalog all of the specimens that we have, make them publicly available online, so researchers, artists, whoever are interested in these specimens to use them can access the information, what we have, and how important it could be for their research. This is a huge task. Cataloging 13.5 million specimens is not easy. This is a small video of showing how we do it, or not. Let's do that. So this is showing our insect digitalization conveyor belt situation. So they're individually taking individuals pinned insect specimens and putting them, the quality is not equal, but you can see it there. So you can put them in, they will be photographed, so the scientist can look at the photographs themselves and not necessarily the physical object. And then all of the label information, the collection information is also photographed and typed out into a database where we can access it online in a findable manner, accessive manner anywhere in the world without actually having to go into the cupboard and taking the specimen out and finding out this information. So this is a huge thing in the museum scene right now, and it's not complete yet, but most of it, the easy stuff is now done. So if you go into our databases, for example, in the Finnish Biodiversity Information Facility, Laji.fi, this is where the data warehouse that we have all of this information available. And you would be surprised how many IT stuff we have. So managing all of this information, be it label information, all of the information that we have from these specimens, curating them in this digital format, accessible for everybody, requires a huge IT effort in a sense. So this is also now known as digital curatorship. So we have this in-house data warehouse, collection management systems, so on and so on, which are also feeding into the global. This bottom here is a database called GBIF, Global Biodiversity Information Facility, which collects biodiversity information from museums and scientific studies all across the world, freely accessible to everybody. So we are hitting a new era of information biology and data-driven, big data ecology. So, what do we do with the collections? This is the most important and classical way we use collections these days. Systematics and taxonomy. And this is the kind of foundation of why the collections were built in the first place. Systematics and taxonomy really tries to answer the question what species are, why is this plant different than the other, are there different species, and what should we call them? It's quite fundamental when you think about it. If this plant will kill you if you eat it, this plant doesn't. And also understanding the biological diversity that we have around us. And how this works in practice is by describing new species, looking at old material to see how the species definitions change and how these species are described and characterized, and then giving them names. And this nomenclatural issue, it's not trivial what a name species has and how do we set the boundaries between different species. And the way we do it is using type specimens. So type specimens have this usually this red label here, and this is a physical representation of the scientist who described the species, what it looks like. There is a description written in some scientific publication somewhere, but nowadays there always needs to be a specimen, a physical specimen that somebody can go back to and check and see what this species describer actually was thinking about. And hosting these important type specimens is a huge important thing for biology all around the world. We have about maybe 250,000 of these type specimens in the collections here that are fundamentally important for safekeeping and the stability of this nomenclatural system of species. And of course, international collaboration. So no one can be an expert in all organisms, and no museum has an expert in every little group of organisms. So we hugely do international collaboration, where museums are sending specimens over and specimen information over using these online databases and physical objects as well to characterize the diversity around us. And then finally, we can use these specimens for ecological studies and other studies. And this is where my PhD comes in. So I'm an ecologist, and we are interested in birds as dispersal vectors for organisms. So basically the main question is, can birds act as lichen taxes? So in ecology, we try to understand how the nature works, and important part of how nature works is how organisms move from one place to another. And if you don't have legs, if you are a small fungus or a lichen, it's not really easy to move around. Or is it? So we tend to think that these things are reproduced by spores, and the historical kind of way of looking at this question is that everything that produces spores is aerially dispersed. Spores just travel in the wind everywhere with wind currents, and everything is everywhere in the end, and then it just falls into a suitable location and grows. But nowadays we have heaps of evidence telling that everything is not everywhere. It is not that simple. Maybe some of these sporulating organisms have other ways of moving around. These are a few examples of the evidence why we think this is. This is a global distribution of this one fungus, and if everything would be everywhere with air, you wouldn't expect this patchy distribution pattern here. And then also morphological evidence. This is a pin lichen. This is a group of lichens that I mostly work with, and they have these stalked fruiting bodies, and on top of the fruiting body you have this black mass of spores. So instead of shooting the spores into the air, like many lichens do, they actually collect the spores into this sticky mass on top of this fruiting body, which seems quite a bad idea if aerial dispersal is what you're going for. So what are they waiting for? We think that they are waiting for an animal to move past and have the spores attached to the body of the animal, and then being transported to a new location. We didn't really know about this animal dispersal thing in small things. We know that they disperse plants and things like that, but with sporulating organisms, the question really was, can birds disperse lichens and fungi? Spoiler, they do. We published this a few years ago now in my PhD, focused on woodpeckers. They seem to be quite a nice fit. They are intimately linked with dead wood, which is a habitat for many threatened species of lichens, for example. And I was planning this research, and I was like, I need to go to the field, I need to catch woodpeckers and do all of this study. And then I realized Natural History Museum collections are there. I don't have to go to the field. The specimens are already collected for me. So I went to the basement of the Natural History Museum and sampled all of these birds. I extracted biological material from them. And this is what I find. And if you start to zoom in, you can find lots of different diversity of biological material attached to the birds. These are extracted from woodpecker feathers, woodpecker feet, and there is a specimen of exactly one of those real woodpecker specimens that were used in this study. And there are some microscope slides that you can go and have a look at this material in situ, the real stuff that was used in this paper. These are different spores of different kinds of fungi. As you can see, there's many different kinds. Here are also particles of lichens on top there, and some free-living algae here as well. So we also see lichens, we see algae. They are green, they have chloroplasts in them, so they're photosynthesizing. We have moss fragments, liverwort fragments. The ball on the top, that's a moss spore. So this is relevant for bryophytes. And then these are diatoms, so they are tiny little algae-like things, like planktonic algae classically. I don't know how did they come up into woodpecker feathers. Maybe the woodpeckers went for a swim, we don't really know, but they're there. And then some animals as well. So these are rotifers. They are classically thought to be planktonic organisms. They live on soil as well. So I also find these nematode worms, and this bottom corner, this creature. This is an amoeba. So you think that amoebas are moving like this, and these blobs of stuff. This is also one of those things, but it's called a testate amoeba. So it has a shell. So it makes this brown little house for it. There's a hard shell, and the organisms, the amoeba lives inside the shell, and then to move it oozes out these pseudopodia tentacles out of the shell that you can see down here. So this is again another group of organisms that we find in these woodpeckers. So woodpeckers seem to be quite relevant for a very huge variety of taxa and organisms that are moving with them. And then finally, what I'm doing now is going further. So pictures of spores will not take me too far on the identification of the organisms that are moving. So I'm using now something called environmental DNA on trying to identify what species of fungi are moving with these birds. So now I get to go to the field and work with bird ringers. I take live specimens or fresh specimens from bird feet, and I extract the DNA out of them, the fungal DNA. And using that, we can identify molecularly what species of fungi are moving with these plants, these birds, and then producing a taxon list of what species are interacting with birds in this way, and then look at the ecological patterns of that. I'm about here in my PhD, so maybe in 6 months' time or something like that, we'll have something published to see what species are actually moving. But with that, I hope I've given you an introduction to the natural history museum scene, and then some inspiration of how specimens can be used for study. I thank you for your time, and I'm open for any questions now or later in this day. Thank you.
Question.
Of course.
Can algae or other small organisms keep living? Like, is there stuff living on that bird?
Why not? So, in general, lichens and things like this, when we collect them, we tend to think that they die. But of course, there probably is viable biological material up to some sense in there. So probably on that bird, it's been collected maybe 10 years ago. The biological material actually living in there is probably a contaminant or mold. We also have, of course, we have living biological material covering every surface in this room, for example. But we would expect that most of the material is dead. You can get DNA out of dead material, of course, as well. But with lichens samples, for example, lichens have the algal partners inside the crust, so they are these symbiotic organisms. So I can go back into the herbarium and rehydrate some of them, and they might pop up back to living. Yes, exactly. Please.
So, thanks. I have a question. So you come from Natural History Museum. I would be interested in hearing how do you negotiate the borderline between nature and culture. For example, what kinds of genetic curating you consider as natural processes, and where does it flip on the cultural side? Absolutely.
So this whole interplay between kind of humanities curatorship and natural history museum curatorship, there is no clear definition or like a boundary between there. although many natural scientists want to think that there is, but of course there is not. I think nowadays with the Natural History Museum scene, we tend to be quite sensitive on how we treat some of this material that also has this kind of humanities or cultural and social effect, and trying to incorporate that as much as we can to what we do in science, but also curatorship and so on. And with genetic material and things like this, nowadays we have quite a lot of international discussion and legislation and things like this to safekeep this cultural and social aspect of information, be that physical or genetical information. So something, for example, like the Nagoya Protocol that was put into place 10-ish years ago is designed as a global initiative to safekeep the use of genetic material and biodiversity information in a way that is fair and equitable across the world, especially looking at indigenous cultures and countries who are biodiversity rich, but then not maybe Natural History Museum rich. And of course, you are aware, and we are aware, that some of this Natural History Museum scene has a troubled past, looking at colonialism and things like this, and it would be wrong to think that the Natural History Museum scene is ignorant of that part. So there is now lots of discussion of dealing with how can we use these collections in a way that is socially just and ecologically and genetically and socially sustainable. So there is definitely movement forward to that, and part of it is, of course, that we are open for collaboration for many different kinds of social and artistic and cultural studies, like this event, for example. But yeah, I could talk about this for hours, so we can have a chat about it later maybe. But that part is integral in what we do and how we manage these collections these days.
Okay, can I just continue? Partly you already answered, but let me phrase it in a different way, because I happened. Well, first, thank you for a very informative talk. But I happened to listen to a book publication event, which was a book by Prudence Gibson from Australia, who studies herbaria in Australia, and the book is called The Plant Thieves. And what I heard was very provocatively exactly how the collection in Australia was made, and how it was made with the help of indigenous people, but those indigenous people and those names of the original plants were not omitted, and so on and so on. So I haven't read the book, but you referred to this already in your mention. And so my question, and also in your talk, you said that there is a lot of information. You showed this the image with the expanding notion of the specimen. So I thought about not only that things can be extracted from the examples, like on a DNA level, but what has been attached to them, and who attaches, and at what level. So do you have an example from the Finnish context? Do we have any of those problems? We like to think in Finland that we have nothing to do with those problems in the big world, a little bit trouble with the Sami, but basically we are so clean and innocent. But are there some examples, sort of problematic cases here?
I fully salute with that, the addition of information, and this has also been, of course, done in many places, that we attach information. It's all about the question of power, of course, and who has the power to do these decisions and attach what information and give out what information. I'm trying to think of an example. I'm a researcher, doctoral researcher, a junior one, so I don't day-to-day work with these kind of issues. But naturally, of course, with specimens coming in from historical times from all around the world, these as you say, Finland, we tend to think that we don't have a colonial past and all of that, that we have a free from jail card because of this. But this is actually not the case, because these international collections and historical collections are, of course, naturally, inherently historical. We have huge material from all around the world, so we are not exempt from this discussion. I cannot think, because I don't work with these directly, so I can't think of an easy example right now. I will probably try to find you later on and discuss and figure something out. Yes. Thanks.
Kiitos, Niku, for a really, really informative and kind of the start to, these kind of issues and grappling with these types of things.
Absolutely.
Is I think something that's going to also be extended in Zayan's take, which is not the next one, but the next one, Aneta. She's looking at an Agapanthus specimen that's actually sitting in the Lumos herbarium, and kind of looking at it from kind of an indigenous angle. So I think, that extends kind of the conversation, but kiitos paljon for really very, very interesting and informative session.
Thank you for having me.
Okay, great. Curator hat on again, and kind of a bridge between Nika's session and Zayan's session. Yes. And once again, bringing in the Tiller Museum objects in their absence. Short interval, objects. A model of CSPR's hospital rheumatic hospital expansion plan, and a stereotactic frame designed by Lauri Lautinen. A copy of Darwin's Power of Movement in Plants. Tiller Museum object, SPR's hospital miniature model, SPR's hospital rheumatic hospital expansion plan. Assembled from plywood, cardboard, metal, wire, and mesh, a miniature model of SPR's Tiller Hospital's unfinished rheumatic hospital extension. The buildings are plywood or coated plywood. In the foreground are trees made of iron wire and netting. Rheumatic disease is an umbrella term that refers to arthritis and several other conditions that affect the joints, tendons, ligaments, bones, and muscles, and because of this, influences the capacity of movement in an individual. Rheumatic diseases like osteoarthritis can lead to severe joint pain from the breakdown of cartilage, the firm but soft tissue that protects a joint when not managed well. At the beginning of 1988, Marina and Yulei ended their 12-year relationship. On 30 March, they set out to walk the Great Wall of China, starting at separate ends in a performance called The Lovers. After 2,500 kilometers, they met on the 3rd of June and said their final goodbye. Seed of Change is a 20-year investigation into the hidden history of ballast flora by the Brazilian artist Maria Teresa Alves. Before the advent of steel ships and the widespread introduction of water pumps in the mid-20th century, heavy materials such as earth, sand, rocks, and rubble were used by merchant vessels as ballasts to counterbalance the weight of their cargoes and crews. In order for boats to take on more freight, this ballast waste was dumped at foreign ports, where it would be reused as landfill. Exotic plants from all over the world flourished on these grounds, altogether altering the landscape. The botanist Dr. Healy Jutila, an expert on ballast flora, writes, "Although seeds seem to be dead, they are in fact alive and can remain vital in soil for decades, and even hundreds of years in a state of dormancy. Seeds contained in ballast soil may germinate and grow, potentially bearing witness to a far more complex narrative of world history than is usually presented by orthodox accounts. Alvis works with a team of historians and scientists to trace these botanical trajectories and uses this information to create floating gardens that represent these foreign species and exhibitions showing paintings, text and maps to further convey these historical entanglements to the public. One of the results of the colonial process was to attempt to break the indigenous relationship with and connection to the land and to dismiss the value of indigenous knowledge of plants. Under colonialism, plants were renamed and reclassified, and many aligned with the white identity. Through digital and live platforms, this project aims to explore the conversation in a way that allows many individuals, particularly those from indigenous and African backgrounds, to share their memories and emotional connections with particular flowers. Recognized as the first female photographer, Anne Atkins started producing the cyanotype that would make up photographs of British algae, cyanotype impressions, in 1843. An apple from Isaac Newton's tree, cross-sectioned. The National Fruit Collection description reads, originated from a tree growing in Isaac Newton's garden at Woolsthorpe Manor near Grantham, Lincolnshire, in 1660. It appears identical to Flower of Kent. Fruits have soft, coarse-textured flesh with a sub-acid flavor. A very shy cropper. A stop-motion video of 2 birch trees growing, compiled by University of Helsinki PhD researcher Sampo Muranen. One of them, on the right-hand side, has an active gene, LASY1, which affects the plant's response to gravity, resulting in the branches growing downwards instead of upwards. Newton's law of universal gravitation is usually stated as that every particle attracts every other particle in the universe with a force that is proportional to the product of their masses and inversely proportional to the square of the distance between their centers. The publication of the law has become known as the first great unification, as it marked the unification of the previously described phenomena of gravity on Earth with known astronomical behaviors. As part of the PIPS in space research, seeds from Isaac Newton's apple tree were sent by the European Space Agency into space to the International Space Station on the 2014 and 15 mission. These seeds floated in microgravity for 6 months before returning to Earth in 2016 to be raised into young trees. And that's the end of my second interval. And I'll just stay here and introduce the next speaker, who is in South Africa in Cape Town at the moment, so she couldn't be with us here, but she recorded her session for us and sent us the video. And she's a good friend and colleague and a very, very inspiring artist that I look to again and again when I want to learn more about myself. And Zayan Khan chose Agapanthus specimen that is situated in the Lumos Herbarium. And she does her object study about this Agapanthus specimen. So just to introduce Zayan for you. Zayan Khan works through seed, land and food from a multidisciplinary perspective, forwarding socio-political, ecological and spiritual political perspectives. Her research is based on interdisciplinary practice, both as practitioner and facilitator. And through storytelling, she attempts to unhinge our dependence on neoliberal consumption. Khan is currently a PhD candidate at the Environmental Humanities South, University of Cape Town, South Africa. And Zayan was going to share one of her works, which is a cyanotype that she made, I think of Agapanthus in her garden, but the cyanotype washed out. So unfortunately, if she said she was going to try to do another one, and if it's possible, she does these gifts with the cyanotype. If it comes through, it will be projected onto the back wall. But alas, if it's not, then we can still dream about how evocative and poignant it would have been. But without further ado, here is Zayan sharing her practice with us.
Hello everyone, I'm Zayan Khan. I'm coming to you from Cape Town, South Africa. And I'm going to tell you a little bit about my work, what I do and how I do it, and then we'll go into the presentation. Thank you so much, Nina, for this opportunity. I do hope that this brings enough fodder amongst the other guests and speakers to think through this topic, as well as how we relate and it relates to various objects. So my work, in the best way that I've been able to figure it out, it's quite an emergent process, is that I work as an artist, deciphering and uncoding different stories in different disciplines to create and become more amenable stories, and stories that are much more easily understood, in order to elicit a sense of agency and sovereignty to the listener, or, you know, within each of us, to somehow create a sense of oftentimes hope in a difficult and sometimes very dark world, to encourage a sense of, you know, transformation. And I use many different tools to do this, in order to do this. I'm very sorry also if my video quality is not that great. I've tried this on different devices. So far, this is the easiest and best way to do this. And ultimately, it's about the story. And so how I do this is I often work from a socio-political perspective, and look at the various ways in which our histories, our traditions, our cultures are kind of forced into boxes, encouraged by many times very capitalistic approaches, and ways of being that encourage separation and encourage individualism, and try to see and oftentimes experiment with ways that weave togetherness and weave ways of doing things, even if it is just a little bit different, just to elicit some kind of response, just to elicit some kind of, if we're able to do things just a little bit different, if we can unfurl the edges of our ways of knowing and our cosmologies and our actions and our habits, perhaps there's a way to encourage, you know, kind of new matter to form and new ways of being to emerge. I'm very encouraged by the way that often small, simple actions can have very grand and big responses and results, but also the small responses and results in a very kind of chaos theory way. And one example of this is through my work, I used to work for a land organization, and in South Africa, because of our many generations and years of forced removal through colonization, through apartheid systems, and now in a very kind of neoliberal capitalistic way, our relationship with land is so electric and so political. And many of us are multiple generations of multiple forced removals. And so thinking through how that affects us, not just in the obvious displacement ways, but also in the ways that our ways of being, our ways of knowing, our epistemological constructs are severed or are disconnected. And what does it mean to reconnect? What does it mean to heal those relationships? And an example of this is in 2012, I learned about the seed laws that our government is pushing very strong within our country, within our region, and within the continent of Africa. And learning that, you know, a lot of these laws and a lot of the ways that these laws need to be ratified and play out within our systems can be, you know, extremely violent, but in very silent ways. An example of this is, so in 2012, learning about the seed laws and what that meant for farmers' rights, and not just farmers' rights, but anybody who uses seed, and how devastating that discovery was for me, because our relationship with seed and our belief systems around seeds and our cultural connections with seed are so ancient as to be something we can consider as a co-evolutionary relationship. And so learning in this workshop that we were sitting with the local University of Cape Town, and various other organizations that were working, kind of land organizations and farmer organizations, justice organizations that were working in these regions, and sitting and kind of absorbing this information and just being so struck by how ultimately cruel these laws are and how they protect very few people who have a lot of money and power. So these corporations ultimately, and people privileged to be working in breeding and businesses essentially, and seeing, I mean, the personhood of a business over the realities and the personhood of, you know, the most rurally poor people within our nation struck me as so rude. And it would be to the point that in many cases, this patented seed was seen as superior to the traditional seed that people had kind of, you know, lived with, and not kind of, that people have been living with for generations, many generations, that this traditional seed had such specific and particular knowledge, in situ knowledge of local climates, local conditions, local environments, local cultures, local recipes, local relationships that could not be bought anyway. That was so specific to each region and each cultivator and each breeder in that, you know, traditional environment, that, you know, this traditional seed is able to adapt and move and be so, what is this word, viscerally strong, resilient to any nuanced or huge changes that may occur within that environment, and understanding the recipes that people have been using for generations and working with that, working within that, working around that. Whereas this kind of laboratory bred seed was not that. The seed knew about, was trying to know about ex situ conditions, but it could never know that because genetically, the seed did not get the opportunity to adapt and respond to the nuanced changes or the huge climate changes within environments. And we've seen it over the last 20 years that the majority of seed that is, you know, purported to be drought resilient or whatever the case is, fails drastically. And when we're talking about the difference between seed and food that is to heal us and to feed our children and feed our families, as opposed to food and seed that is for sale and is for yield and is for income and capital, basically, where the intentions and the our expected results for them are so vastly different. Even though it is talking to food security, it's a very thin narration or a very thin storytelling of what food security is, that it is incomparable. The two are incomparable. The one which is so lauded and so supported, doesn't work, and we've seen it time and time again, doesn't work in those, and this is a grave generalization, but to get the point across, doesn't work in the environments where people are, responding to the needs of their, pregnant women and feeding babies and people who are ill, and understanding how to make the food last through the season, and having those kind of knowledges, and the violence in that, as an example of the violence in that, is that oftentimes what would happen with the extension service officers, so people who are working for government, the agricultural officers, would go into villages, and I really hope this doesn't happen anymore, because we'd heard this story time and time again when I was working for the organization, and we lobbied government, and we made a big noise about it. Is that these extension service officers oftentimes would go into communities and confiscate entire seed libraries that had been grown over generations, and replace them with these bred varieties, which, like I said, often didn't work because they didn't understand the nuance of these villages that people have been living in, and rural landscapes and those relationships. Often people will say that the true value or the true wealth of their community is in their seed library. So when these entire libraries were confiscated, people's food sovereignty, people's medical health sovereignty, all of these things were taken away. And there's this whole rhetoric around how corporate knowledge or new knowledge or university knowledge is so much more important and so much more better and so much more knowledgeable than traditional knowledges, even though a lot of them are based and are taking the knowledge, like with seed, they're taking seed that exists already, that people have bred and cultivated over generations, of millennia in often cases, and improving them without actually improving them, and creating kind of this huge negative impact. So at the end of the day, or end of my little story, is that hearing about these stories and understanding these ways of being and knowing, it is my duty and my job to share these stories on a wider platform, which I do with the seed story in specific. The solution that I came to for myself was to create a seed library, a seed bibliotheek that tells the stories of seed, and not necessarily around, so it pivots on the story of seed instead of on how many varieties we have, or the quality of seed, even, because we're very open to accepting seed no matter what the seed is, because it's linked. Like, people, when they donate seed, when they share seed, there's a huge story connected to that. And so having that, understanding that story becomes really important because those are our cultural connections. Those are the value systems that we are wishing to push into the future, where stories are being shared with our children, stories are being shared with new people, stories are being shared with each other, especially around the ways that we struggle, and especially around the solutions that we have. So that there's a sense of interconnectedness and a sense of community driven by our experiences that we know work or don't work. And an example of this is, for example, when we first launched the library, the seed library, somebody had donated tree seed, indigenous tree seed. So perfect, wonderful. We love indigenous seed. But it was 40 years old. And it struck me so much that someone could take this seed that they had harvested 40 years previously and care for it for so long, even if it meant it was in a drawer in the back room somewhere. The fact is, the seed was kept, and the seed was kept, it was well kept, well stored. And we thought, well, what is the meaning of this? What do we do with this? We can't say no thank you. And so we realized that because of that care that someone had taken to, first of all, hold on to the seed, maybe they inherited it from somewhere, but that they carried it to us, that they thought, here's a space that will understand this. We decided we'll accept the seed. We did a germination test just out of interest, and literally 3 seeds, 3 seeds sprouted. And the rest of them, we decided, because they were very hardy seed, we decided, let's wash them and make an instrument out of them. And so it started this discussion and the story about how we use seed, and in the various ways that seed are kind of tools within our lives as part of the story of co-evolution, that we use seed for jewelry, that we use seed for making color, that we use seed for teethers for babies, for making musical instruments, all of these different, not adaptations, but all of these different ways that we use these tools. So we thought that was very powerful and very, and also very fun. There is this element of joy that we have to work with and we have to elicit in order to encourage us to carry on doing this kind of work. Okay, so that is, a very long introduction to what I do, but hopefully it tells a bit of the story. So onto the topic of what I'm bringing to you today. One of the experiences that has really kind of pretty much shifted my state of being, and I'm sure this happens for almost everyone, if not everyone who enters into this, is that I am freshly 4 years into being a mother of 2 boys, and because I'm not sure what the culture is locally there of having children, but here it's not that common to have children later on, but so to kind of go through life and then have children in my late 30s has been a very jarring experience, which I love and welcome, and there's a lot of joy, but it's very jarring. And it's recalibrated my way of thinking in a lot of ways, and often mostly in very good ways. But what shocked me so much was how difficult it was to be sovereign in the state of pregnancy and birthing and postpartum, early postpartum. And what shocked me so much was how usurped, how taken up into allopathic medicine, specifically pregnancy, was. And because of my nature and my kind of healing modalities, the hospital was never a place that I felt safe for various reasons, and allopathic medicine wasn't really isn't really my first kind of port of call for care for myself or my family. And so I found it so peculiar and so strange that it often was the only way of medicine that people sought out, especially the generation above me. So when I shared with my mum and my family that I wanted to have a home birth, this was very scary for them. And my mother pleaded with me, please, especially your firstborn, do it in the hospital. Do it with people who know what they're doing. And I found it very, there was this kind of big dissonance in this big moment where I just couldn't understand how that had come to be. And I couldn't understand why that was really one of the only options, because we did not have medical aid, and so it was a very expensive endeavor. And I didn't really want to go into the public health system, and I would have if I needed to, but it's also not a place that I feel particularly safe. And there's a lot of violence that happens, and I just heard too many stories. Long story short, we decided to go home birth. We had a team of midwives, and I began to learn as much as I could. However, I did not have any previous experience of having this journey and having this relationship of, growing human inside human. And it was confusing. It was, I was at a loss many times. And I would often ask myself, because of the state of my pregnancy, am I sick, or is this just a symptom of pregnancy? And that became kind of a mantra over the months. Like, is this supposed to happen? Am I supposed to feel this? And luckily, one of my dearest friends who had just had a baby was busy studying traditional Chinese medicine with her dad, who is, 10 generations of Chinese medicine practitioners. And those of you who are scientists, or those of you who kind of work in medicine or in similar fields, will understand the importance of generational knowledge, scientific generational knowledge, that is embedded in so many generations. And their particular field of medicine is around women's health, babies, so this postpartum, this care, not just in that time, but how that affects particularly birthers, so let's say women, birthing people throughout their entire lives. And so I felt safe speaking with her and allowing my symptoms to unravel and unfold. And one of the things that kept shocking me was, in my very mixed heritage, which includes, not to base it into this, but genetics from this land, it also includes genetics because of our colonial histories, from parts of Asia, parts of Europe. And so I know, there are parts of me everywhere that come from everywhere. But I was so struck that we didn't have a lot of our own traditional knowledge around birth in my direct lineages. And I was seeking knowledge out of Chinese systems, which, yes, I have Chinese ancestries somewhere in there. That's my son, excuse me. I have knowledges. I'm so sorry about that. And I realized that I'm running out of time. But so struck by where our stories were that I started to investigate. Long story short, I became very ill, extremely nauseous, deeply fatigued, very confused as to why this was a way of being while you were pregnant. And I ended up seeking out the knowledge. They made me very strong teas, which helped me tremendously. I felt stronger and more creative and better than I did from even before I was pregnant. And the birth at home was excruciating. Labor was intense. I ended up being in a 36-hour labor, and that was really majority because I had never experienced it before and I had no tools for the pain. We fell pregnant soon after that when my son was 13 months old, and we decided to go ahead with it, but I was determined to do it on my own, in my own ways, so that I could feel what that sovereignty was. I studied very hard. I ended up carrying full term. So the baby that you just heard, this is the baby that I'm talking about right now, and was very well prepared with a lot of medicines, deep spiritual practice. And one of the best tools that I used for that, which is linking to the story of trying to revive and retrieve indigenous knowledge, traditional knowledge around birthing, was the use of agapanthus. The roots which I dried, boiled, and drank as a tea in the last trimester of my pregnancy, which I tried to do in the first birth, but I didn't dry the roots out. And so it was irritating for the throat, and I knew that as soon as I drank it, that, I mean, I have also history of gently, this is the wrong word to use, but gently poisoning myself while seeking out, taking the threads of knowledges from different researchers and books and conjuring recipes from them and solutions from them, which is something I don't recommend doing. It's something that I do myself, and it's not something I do irresponsibly. But understanding what those gentle ways of practice are. But so I knew through research and, through the work that I'd done and the experience that I'd had, that agapanthus needed to be dried before. And this wasn't written anywhere. This is something that I had to figure out. But because my first language in work, let's say, is plants, I understood that about them. Agapanthus is also something that I've cultivated for many, many years. So we have an intimate relationship. I was gifted some agapanthus that grows in the Storms River mouth, which is a local river estuary connected to the sea in the region. And what's very interesting about this agapanthus that I used is that the roots were literally dangling in salt water, which is not a normal condition for them. They don't tend to work well in salty conditions. So this plant had adapted to this, strongly adapted to this environment. And why that's wonderful is my garden has got a salty nature. We're close to the sea and we're on a flail, we're on a body of water that has, this kind of salt richness. And I understand the traditional way of knowing about agapanthus is that it's a journeying plant, and so because of the flowers, kind of the signature of the flowers, there's a like as Liliaceae, it's got a tubular opening, so it does kind of this trumpet shape, and that is understood to be a journey, a transition. And when I went into labor, the pain, I was in denial about it for basically until the baby came out. My water didn't break or anything. I was having what I thought were Braxton Hicks contractions. But I experienced in the surge and the pain of the surge, I experienced and I felt and I saw in my mind's eye my cervix dilating very quickly, very rapidly, which we know happens, even though midwives will often tell you, which happened to me in my first birth, that you're not dilated enough. So stop the fetal ejection impulse, which was so damaging to me. But I visioned this and I saw it. And what was wonderful about being alone and not laboring with people telling your body what to do, and having your own power to do it, and not handing your power over to someone else to tell you what your body is doing, was that I could just let it happen. And literally, that experience, birthing my baby, was 3 hours long compared to the 36 hours previously. A healthy baby, a healthy mama, it was a really glorious experience. And there was in that doing much healing that had happened, not just from the first birth that I had experienced that I found very traumatic, but from the reconnection and ways of being sovereign in our birthing practice, that it's not that difficult to revive and reclaim those knowledges and ways of being. It's hard work, and it's access to knowledge, and it's the desire to seek it out. But now that I have that experience, I understand so much more about that world. And that is to say that we see pregnancy and birth as a form of dis-ease. Obviously, it's a heightened stress environment and situation, but the way that we objectify and we pity people going through it is something that's very damaging, personally, I found. And I was happy to release my sovereignty and release my agency, which was one of the worst things I could have done. But I'm grateful for that experience because of how much it has taught me. I have gone over time now. I'm going to leave it there. And I thank you very much for this opportunity.
Okay, so we can unfortunately not ask Zayan any questions. And I'm cognizant of time. And what I thought I would free play here to the distraught of the technical team. It's not bad, I promise. I was thinking that I might just open the floor, maybe just for if anybody feels they want to make an observation, because Zayan's not here to actually speak back to it. But if anybody has observation they want to share with the audience, we can do that. And then I want to invite Vincent Rockmanac to just introduce, again, sorry for the team, just introduce the workshop that we are doing this afternoon. And I'm going to introduce Vincent as well before he comes on stage. And then my ending is not another PowerPoint. My ending is going to be time for you to, just before lunch, spend time with these tables now that we've had a whole day. Sorry, Sampa. Now that we had this whole day of presentations and talks and breathing exercises, for you to kind of carry those resonances with you as you now look at the table again and the relationships on these tables. Hopefully you all are around at 5 o'clock to look at the outcome of the income of the workshop. But yeah, I hope that suits you, because then we might just be on time for lunch. I don't want to keep you too long. Are there any observations in terms of Zayan's presentation that anybody just feels they want to share in the room? If not, I just wanted to create the opportunity for people to share something if they felt they wanted to. Oh, Aneta.
Well, thank you. But I just wanted to share that the agapanthus is a beautiful blue lily, so you can go online and look at it. I don't know what it's called in Finnish.
Yeah, and the specimen is actually on the table, so you can have a look, the one that she responded to. It's on there, the dried specimen. Collected in 1982 in South Africa by a Finnish botanist. So yeah, it's over there. Was there anybody else? Okie dokie. So, Vincent Rockmanac, for those of you who don't know. Vincent Rockmanac is a Helsinki-based Basque-French discipline fluid artist and researcher. Vincent started his career in theatre as a director, but gradually moved away from straight theatre context. Instead, he has been focusing on how the concepts of stage and theatricality transform in the context of climate change, technology, and transmediality, at the crossing of visual installation and performing arts. Exploring new artistic ways to engage with contemporary, multiple, and complex environments. In 2020, Vincent completed his doctorate in arts at UniArts Helsinki with his research project,
I know.
The stage, sorry, it's the end of the day now, I will fall over my words. After that, he began the 4-year postdoctoral artistic research project, Data Ocean Theatre, as a visiting researcher at the same institution. And I'm so very happy to have you doing this workshop, and very privileged to feel it and to share in terms of the art making and research space I can vouch for the fact that you have taught me that art can indeed still elicit wonder in a spectator, and the power and the force of that. And it's a wonderful thing to be, to get my faith restored in that. So, welcome.
Well, I thank you, Nina. I'm blushing here, like matching my pullover. Thank you very much. Actually, I grew up. My father was a gardener, and I grew up in a flower shop. And actually, my grandfather was growing plants and trees. It was the owner of a tree nursery to reforest, and one of the species that was grown in the nursery and sold in the flower shop was a agapanthus, and it happened to be one of my favorite flowers. So, yeah. Yes. So Nina proposed me to facilitate a workshop that would put together artists, researchers, and scientists, and it's based on, I think, that Nina happened to know my work that I did based on my dialogue with the Marine Research Unit of the Finnish Meteorological Institute. That was the first step in the Data Ocean Theatre project, and I met this research unit, the Marine Research Unit, so investigating Baltic Sea changes, mainly under climate change conditions. And we started a dialogue, and there was a concern by the Finnish Meteorological Institute team that they were saying, like, we have like a lot of data visualization, but we feel that we don't reach out with the abstractness of those scientific data and scientific data visualization. And they asked me, can you do something out of them that they might reach out differently? And I say, yeah, well, yes, that's an interesting thing to shift from the data visualization into artistic materialization and to ponder, does it produce another effect, that it sensitizes differently the people who are receiving them, the public. So actually I started to work with data visualization from the Marine Research Unit in terms of temperature changes, salinity changes, surface changes, and I turned them into artifacts, art objects that I put then into installation, into performance that were part of my last work and exhibition that was presented last month in Moving in November Festival in Kiasma Theatre, on Kiasma Theatre stage. So the workshop shortly, because it's soon lunchtime, and I don't know if you go to the cafeteria, but today is Christmas lunch, so it's special lunch at the cafeteria. So the idea is that we work from data visualization from scientists involved in Nina's research project, which is, we have to say, which is going through several months. So that's one session among several sessions of work between those different researchers. And for two chosen graphs, one is from Yasmin, and I think we saw it in the morning, so that was really nice to see it pop on the screen. We will invite groups of people made out of artists and scientists to collaborate into creating artistic materialization from those scientific data. Because it was about, I forgot her name. Zan, the last speaker, Zeon, spoke about joy and the pleasure. So, and to finish the Scuba Research Days is also like to kind of make to cycle the cycle also with the first day on romantic irony and playfulness. So the idea is to go playful and joyful with that work. So hands-on, making kind of making happen a prototyping factory when with a little amount of time we try to make things to appear in the space. So from 2 graphs we're going to make objects that fall maybe upon a kind of aesthetic regime that I am bringing and proposing myself, meaning theater, carnivalesque, costume, mask-oriented objects that at the end of the day will be displayed as an installation or a mise en scène or we don't know what yet. So yeah, that's it. And we're going to have a little presentation. We open the doors of the workshop at 5, from 5 to 6, to gather people to share the outcome, to discuss and to celebrate the end of those beautiful 3 Couva Research Days. Did I forget
Merci,
something?
merci. No, that's perfect. That's it from me for the first half of the day. I hope there's a lot of conversations to be had. Hopefully that extends what's happened here. Resonances that continue to build. But yeah, kiitos for being here in a Mercury retrograde situation. And we hope to see you at 5 o'clock. The venue is, for those of you who don't know, is it the 6th floor or the 5th floor? It's the 6th floor, right? Yeah, the 5th with a lift, and then there's a door and a spiraling staircase that you need to go up. But I will be around on the 5th level, maybe just before 5, so that I can direct you up. Yeah, it's the lighthouse. So you can ask anybody where is the lighthouse, and they will also be able to show you. But kiitos for coming in this very difficult day. And yeah, we hope to see you. Yay! Okay, yeah, kiitos, and thanks for the technical team for being ridiculously amazing and wonderful. Really, thank you so much for that. And Michaela and Mika and everybody else that's helped so much. I really appreciate it. And my speakers, thank you for coming. Really, you made my day, literally. Okay, toodaloo, moi moi, for now.